Okay, so you're good to go.
Great. Good morning to those of you joining us from Australia, and good afternoon to those of you joining us from the United States. My name is Stanley Grant, and I'm a professor of civil and environmental engineering here at University of California, Irvine. I'm also director of the UCI Water-PIRE, which is funded by the National Science Foundation and supports joint research with our Australian colleagues on the use of low-impact development, or LID, or as they say in Australia, water-sensitive urban design, or WSUD, for water supply augmentation and ecosystem restoration. As part of our outreach effort for the UCI Water-PIRE, we are hosting a series of mini symposiums on different aspects of LID research and practice. The first of these, which was held a couple weeks ago, focused on the use of LID for reducing flood risk in urban areas. The second, which occurred yesterday, was on incentives and barriers to LID adoption. And today's mini symposium, the third in the series, will focus on the use of LID for water supply augmentation primarily. We are very fortunate to have really an astounding lineup of speakers and panelists on today's symposium. They are some of the leading researchers in their respective areas. I'd like to start by introducing our speakers, and for the sake of time, we're just going to have them wave to you when I call out their name, and then you'll get to hear from them personally in a minute. Our speakers include Professor Anna Deletic from Monash University and the director of the Monash Infrastructure Research Institute. So give us a wave, Anna. There you are. And Professor David McCarthy, also from Monash University, and an ARC DECRA Fellow and Senior Lecturer in Water Engineering. And I'm not sure if David's online, but I know he'll be joining us in a moment. Professor Sunny Jiang, professor at the University of California, Irvine in Civil and Environmental Engineering and Ecology and Evolutionary Biology. So Sunny, give us a wave. That's great. Dr. Megan Rippy to my right from UCI and a postdoc on the PIRE program at UCI. Dr. Kefeng Zhang at Monash University and a Civil Engineering Research Fellow. Thank you. And Professor Tim Fletcher at the University of Melbourne and professor in the Center for Water for Livability. I don't know if Tim's on yet. And our panelists include, at least at various points in time, the great thing about Google Hangouts is that people can hang out and they come and go. But Patricia Holden at UC Santa Barbara and a professor of microbiology there in the Brin School, I know was going to join us. Jim Murray from the city of Santa Barbara and the Water Quality Creeks Division was also going to join us, especially for the Q&A, hopefully. Brandon Winfrey, a postdoc on the PIRE program at UCLA, and I know Brandon's there. Hi, Brandon. And Andrew Marion, who's a postdoc on the PIRE at UC San Diego. So again, I don't see Brandon at the moment, but hopefully we'll see him later. So the format of the symposium will be as follows. The speakers will give a short 15-minute talk followed by a 5-minute question and answer period. And after all of the speakers have finished their presentations, we will transition into a roundtable discussion and then conclude the symposium. So without further delay, I would like to introduce our first speaker, Professor Anna Deletic, who will be speaking on engineering challenges of LID. And with that, Anna, please take it from here.
Thanks, Dan. Thank you for the introduction. So I hope this works. So I'm going to talk about some key engineering challenges that we are facing in implementation and also design of these systems. So in our second slide, I'm just showing some of the key challenges that we are facing. First of all, construction and maintenance are a huge issue. Industry capacity to build this system is not great, and the reason is that they are novel, they're new, and although they look very simple, they actually do require quite a lot of experience to make them operational and to do their job well. Particularly, the challenge is long-term operations. We don't know a lot about these systems being not very old. We know that clogging and perhaps meta accumulation may be a big problem. So I'll try to talk a little bit about our latest research in particularly long-term operations, because honestly, industry capacity building is an ongoing process which happens not in my lab, but I believe this event is part of that as well. However, there are some design issues. These systems are still to be developed further. One of the biggest issues that we have with WUSUD systems in general is that they take land. In our urban areas, land is precious, it's very expensive, and that's a problem. Developers don't want to give up land. Some people are saying they're not nice looking. For example, biofiltration systems got a bit of bad mouth from some communities because they use only native plants, and some communities don't like it, and people are saying they're definitely not the best looking garden beds. Plants can die quickly because during dry spells, so if we have several weeks of no rain, plants can become pretty stressed, and particularly if we are doing stormwater harvesting, sometimes solo stormwater systems that capture only stormwater don't look pretty effective, particularly during long dry spells, like in, for example, Israel, where I worked a lot. They don't have rain for 7 months. Okay, let's talk about long-term operations. As I mentioned, clogging, which is on slide, I think, 5, we are talking about clogging as a big problem particularly because of surface accumulation of sediment. So on sediment, on the surface, right on the surface, we can see lots of sediment, and if it's fine, it can cause pores to get clogged. Why is this the problem? The problem is because we actually have then lots of untreated water that comes and overflows, and definitely can impact the plant health and their survival. In 2007 and 6 and 7, so it's been a while now, we surveyed 36 systems, and these biofiltration systems were pretty young at the time. And what we found that actually the main reason for almost 50% of them being clogged is that they used wrong media. So although this is an operational phenomenon, it's actually far more about design. And if you put wrong media in, definitely it won't work. We did lots of work with particularly a council here in Melbourne to try to prolong life of these systems and kind of solve this engineering issue. So what we've done, apart from knowing that plants are important, they kind of dig the surface and keep infiltration happening. What we also innovated is that you can put on top of your biofilter a protective layer of coarser media. This media is kind of same as your transition layer media. So on these slides here on the right, you see that we are talking about actually anywhere between 50 and 100 millimeter of protective layer. And that protective layer catches sediment and protects media, which is what's in the middle, from clogging. We worked with this council, City of Manningham here in Melbourne, and we developed something which is called zero maintenance bioretentions. You can see on the left-hand side here that these systems fit within a nature strip. You can see in the middle how basically they're walkable areas. People don't even know they are there, and you see some implemented on the bottom right. What are innovations here? Call it innovations, but it's actually just using a bit of engineering approach to these systems to achieve the goal, which is biofilters which don't need any extra maintenance is that we first have grooves in gutters. So you can see on this slide lots of grooves with sediment accumulation there to trap the sediment before it enters the biofilter. Biofilter, you can see in the picture in the middle, is just behind this entrance. There is this clogging protective layer, and then there is buffalo grass. What is great about this system that they are part of nature strip, what we call in Australia. And in Australia, residents, they have obligation to mow this grass. So this happens without any extra investment from a council. And Manningham actually built quite a large number of these systems now, and we are monitoring and trying to perfect their design. Heavy metal accumulations being raised as a possible problem by lots of people, and we've been always asked how long are they going to last before they become basically full of heavy metals and in a way contaminated land. We've done 2 surveys of 36 field-scale biofilters across Melbourne. First survey was in 2006-07, and the second just 2 years ago. These were some of the earliest systems ever built in Australia, but now some of them are 11 years old. They're very variable in their design, so on this slide here, it actually specifies how different they are from each other. So now I'm showing slide, it's slide 9, which actually shows concentration of heavy metals as surveyed in 2007 and now 2014. And we were very surprised when we saw the data from 2014, because we thought that we'll have all these metals accumulated, and we thought we will have a problem. But for a particular reason, and I'll talk about that a little bit later on, there was a decrease in cadmium level. Copper and zinc did not change a lot, and there was a slight decrease in lead. But more or less, all systems, the same systems that we surveyed 7-8 years ago, were having same level of metals as when they were built, which is quite surprising. But then when we think about how these systems work, obviously, we have plants that are using not just nutrients, but very likely trace elements such as heavy metals to grow. So we are now doing further research to understand this mechanism, but we are very pleased to learn that even after 11 years, these systems retain the same level of heavy metals as at the start. Just to let you know that one biofilter, or the group of biofilters I'm showing here, was regarded as the most polluted at the start of the survey. So all others have much lower concentrations of heavy metals. That's kind of main things I wanted to talk about, long-term operation problems and maintenance issues, which mainly resolve around clogging and heavy metals. The next chapter of this presentation is about how to achieve robust design. I highlighted at the start that we have issues with space, how to solve space issues. The answer is very simple. We're now building vertical biofilters. For example, living walls for water treatment is something that we are developing here in Monash. They look beautiful. Like you can see here, this house which is totally covered in vines, and that shows you how you can actually make quite a beautiful, attractive feature out of a biofilter. So they can be fitted everywhere, and on the next slide I'm showing possible, these are not biofilters These are actually living walls, as they are called, but they can become biofilters. And they can be a part of a very busy street, you can see on the top right, or actually they can start creating noise barriers. And you can see some of our ideas how we can turn biofilters along roads into noise barriers as well as pollution control barriers. So imagine that all runoff from this road goes into such a vertical biofilter which uses vines to grow. So another thing that we are experimenting with are green walls. So living walls are more or less look same as biofilters, but the only difference that you use is vines instead of Carax or Melaleuca. In the case of green walls, you grow plants from a wall. So in your in slide 13, I'm showing actually what we are doing in the lab at the moment, and trying to turn some beautiful piece of art, a green art like this one, into, in our case, green wall for grey water treatment. So we have a guy who's developing this technology where grey water, light grey water, is treated through a wall like this to develop non-portable water. But I won't talk more about that because this is about stormwater. In slide number 14, I want to just highlight that we are trying to solve the problem of aesthetics as perceived by some of our people in Melbourne by increasing the use of ornamental plants. At the moment, we've been experimenting with plants like canna lilies, which are beautiful, and of course some of the vines that I mentioned before, but particularly some flowering ornamental plants which can add amenity value. So hypothesis was that some of these plants are very good for both nitrogen and phosphorus removal, but also they increase aesthetics of the systems. Finally, to solve problems with long dry periods and low efficiency, we're experimenting with developing, we call them hybrid systems, which can treat different types of water, not just stormwater, but we use, and here in slide 15, I'm showing our pilot system in Israel, where during 5 wet months, winter months, we are harvesting stormwater from a local drain, and that treated water is then returned to an aquifer. So that's this first operational mode. But then during 7 dry months, we are actually using the same system to treat contaminated well. This was actually an inspiration for us, and since then, we started developing another technology for which is called living walls for grey water recycling. So you can see on the next slide how a living wall, we can use grey water, which is basically only shower and washing basins, with some minimum pre-treatment. It goes into living wall, which creates beautiful ornamental feature of a wall. And then we store that water and use for recycling, garden irrigation, or toilet flushing. We built the first system using technology that we developed in our lab, and are monitoring how it works in the field. So what we've done is we, as I said, looked lots of different plants, but also played with a novel media for antimicrobial removal. It's some new material we made here in Monash, copper-coated zeolites, which are good for pathogen removal. But the systems more or less look the same, but plants, as you see on the next slide, look quite different from typical biofiltration plants, which we know are effective for nitrogen removal, but as I said, did not get lots of credit for looking beautiful. I'm showing some results. So here is nitrogen, and this is grey water only. So you can see that we have inflows which are much higher than usually found in stormwater, at least in Melbourne. And of course, Carax with novel media and with just normal media is doing fantastic. We knew that. But we are finding that there are a few ornamental plants that are doing as well as Carax. So canna lily, particularly this beautiful canna lily, and also a few climbers are doing really good jobs. So no big difference there. And removal was pretty good. TP for grey water only was a bigger challenge because TP in grey water is actually 10 times more. Excuse me, grey water contains 10 times more phosphorus than stormwater, and obviously that was a challenge. Carax did very well, but actually canna lily was not that bad either. It's not as good as Carax, but it's pretty good in its own right. Finally, I mentioned that, and that was the whole point of starting with grey water, is that we are now developing systems for hybrid operation. So we are using a grey water in dry days and storm water when it rains. So these hybrid systems, we are hoping to look like this one shown in the slide 20. So as you can see, they should be pretty. That's why we've been using the same ornamental plants. And here I'm presenting actually quite, on slide 21, very latest results. To be honest, these results have never been presented anywhere before. We just got them out of lab, where we are showing removal of nitrogen. Here you can see in the inflow, and you can see it's variable because some of these events are grey water and other events are storm water. And again, some for dry weather and some for wet weather. Unvegetated systems here in red, circled in red, are pretty bad. They are obviously not doing anything, and particularly after long drying spells, we are having breakthrough of nitrogen. However, look at our Carax and look some of our nice ornamental plants, particularly canna. They're doing pretty good job in removing nitrogen and even doing fine after long dry spells when we know problems with nitrogen removal is big. So that shows that these hybrid systems, and we've been running this now for 6 months, so these data coming from 6 months of trials, so pretty mature systems, showing that systems which remove stormwater and grey water at the same time, whenever, depends whether it rains or not, are pretty good. TP removal, it's more challenging because the variations are far higher than in the case of nitrogen. Again, unvegetated pretty poor, and vegetated are doing good. So more or less, we are seeing that this is working. The last slide is just showing, actually, not the last slide, the slide 23 is just talking about us. Here at Monash, we have more than 10 years of experience in developing biofiltration technologies. The first biofiltration guidelines were published in 2009, and last year we issued a new version with all sorts of novel things related particularly to stormwater harvesting, but also different plants. Unfortunately, some of the research I just show is not there because it's a very new research. And the final slide, I just want to give credit to lots of people that were working here. These are not these are just people who've been with us for the last few years, but the list of people who worked on biofilters at Monash is much, much longer. Thank you very much. Any questions?
Thank you, Anna. That was a fabulous presentation and a great way to start the symposium. Are there any questions for Anna from the panel or the speakers?
I have a question. You mean speakers today, or?
No, Andrew, you're on. Go ahead.
Yeah, Anna, that was really great. Thanks, sir. That was really informative. And I had a question. You know, I saw that you're using a lot of different ornamental plants, and some of them were not native. You know, some of your good performers are also plants that can be really aggressive, you know, in invading nearby spaces and things like honeysuckle and grapes. You know, they tend to spread out. Is that a concern as far as plant selection?
What happened, as I said, there was a big piece of social study and also social science study, but particularly Stormwater, I think Victoria and Melbourne Water, they ran a big survey, because now we have, you know, thousands of these systems all over Australia, and particularly in Melbourne. And more or less, I was shocked personally, it came out that people are saying they don't like how they look, particularly people in areas where, you know, space is premium. They're saying they are ugly, and they say can't we use ornamental plants. Then we convened a workshop with the landscape architects, and these plants that we are actually now testing and tested are plants that landscape architects are saying are very popular in so-called gardens, you know, ornamental gardens of Melbourne. So you will have these species everywhere, like canna lily, yeah, it's everywhere. Also these vines and stuff. Yes, they're definitely not native. Yes, they're not very likely great for biodiversity, but these are the plants that our city in Australia is full of and is sold in, you know, as ornamental plants. So being an engineer, I responded to the push from our industry for ornamental plants, and as I said, I don't think that rain gardens are going to make a huge difference in the market, which is saturated by these plants as part of, you know, as you know, gardening industry is huge everywhere in the world, and
right.
Me telling them don't use canna. It's, you know, they're doing it anyway.
These are already widespread.
Oh, they are everywhere. Like,
okay.
So that's what I'm saying. I'm giving you a long story why we did this, because and most importantly, these are the plants which are widely used in gardening industry.
Okay, that makes sense. Thanks.
Any other questions for Anna before we move on? All right, great. Well, I just want to say personally, I think that the idea of having hybrid systems that use stormwater during the rainy season and then gray water during dry weather is really excellent, and I think would maybe make sense in arid areas, semi-arid areas like Southern California, for example.
Absolutely, and actually the idea came from our work in Israel. Melbourne does not need this that much, but if you are in Israel, it's really a problem. Also, I know here in some, like Perth in Australia, I mean not in Melbourne, in Perth, that's a huge problem as well. And what we know, and we knew that for a while now, if plants are stressed, they stop working. So it's not just that they will die off, but actually the system after a long dry spell is going to leach nitrogen. It's actually causing pollution. So having some other water types in between is fabulous for so many reasons. Plus, it's actually
Oh.
Looks like Hey, Anna, it looks like you need to unmute yourself.
It looked like David muted you.
And then he dropped off. If you scroll to the top of your screen, there should be a button that has a little microphone.
Okay.
There
you go. You're back. Yeah. Well, I was going to say also, Anna, that you know we have this sort of ironic situation in Southern California where, in order to maintain the biocomponent of biofilters, you know, during the summer period, we would actually have to irrigate them.
Yeah, they're water sprinklers.
So that just doesn't make any sense at all. So
I agree. And that's why we started this in Israel, because they have 7 months of no rain. And then they had these polluted wells of nitrate, with nitrate concentration of 140, crazy polluted. And we said, let's try. And plants just went, wow, you know, they were growing beautifully. And then we started, you know, saying this is a good idea. So as I said, it came from a real need in Israel. And now we are moving to this hybrid with grey water. We are thinking, but honestly, when it comes to beauty and how they look, these green walls, I have not presented any on that because they're pure at the moment, grey water, but they can become also stormwater and grey water, so it can be roof runoff, and then of course grey water, and they're beautiful. They're pieces of art. So that's the newest babies that we are.
Yeah, that's great. Thank you. So Anna will be joining us for the roundtable, and so if you have burning questions, we can come back and ask her at that time as well. So so if I may, I'd like to transition to Professor David McCarthy, who's who's going to talk to us about a really comprehensive study that they did at Monash University, looking at viruses, bacteria, and pathogens generally in stormwater runoff. So, David, I don't know if you've been following how we're running this, but the idea is that you'll just go through your presentation. We all have a copy of your presentation, so we can follow along. Maybe if you give us some cues as you go, you just tell us occasionally which slide you're looking at, and then when this is converted into a course module, we will merge your beautiful face and your slides later.
Lovely. Thanks, Dan.
Okay,
and sorry for muting someone before. Anyway.
Yeah.
So,
as James says, you mute and run. So.
I thought I was only muting it for me because I had a phone call. Anyway, so pathogens in urban stormwater. So the work that I'll be presenting on today is a combination of a whole heap of co-authors. So I've just put mine and Anna's name, but it really extends to a whole heap of people around Australia, and in fact some other people internationally as well. So the topic today is purely pathogens in stormwater. So I'm not covering treatment of pathogens in stormwater, just purely what's in stormwater. So a bit of an outline, so I'm going to the next slide now. A bit of an outline for today is that I'm going to talk a little bit about where we started with all this, which was back in 2006 in measuring fecal indicator organisms in urban stormwater, and whether or not we could predict their behaviour using a very simple conceptual model called MOPIS, Microorganism Prediction in Stormwater. And then we're going to go into some more details around pathogens in urban stormwater, and a little bit about microbial source tracking, because I think source tracking is an important aspect when we're talking about pathogens in urban stormwater. So I'll get to details about that later. So on to the next slide now. So in the early years, as I said, we started with monitoring these fecal indicator organisms. So back in 2005 to 2007, we monitored about 4 catchments, ranging in event sizes, but mainly we've got around 20 events for each catchment, and around 14 events over each, 14 samples over each event, and analyse them for a range of indicator organisms. And we used that data to start looking at whether or not we could explain the variability of these organisms between events. So why does one event have particularly high levels of contamination versus another event at a particular site? And what we found was quite simply that there were antecedent climatic parameters such as vapor pressure, temperature, and relative humidities, which explained part of that variability. between events, but also the rainfall intensities of the events at each site. So we found that two parameters were able to capture a significant proportion of that variability between different events. Some wash-off parameter, which was the rainfall, and some antecedent climatic parameter, which kind of represented the build-up of pollution on the catchment surface before the event started. We then looked at whether or not we could explain the within-event variability of E. coli, and whether or not some simple rainfall intensity metric could explain that variability. We weren't so happy with this, but it was as good as we got. We got in about 15% or 20% of the events, we got very significant correlations between rainfall intensities and E. coli, whereas in the majority of the events, they weren't significant. Anyway, we pressed on. This is the joy of modelling biologically active organisms in stormwater systems. So we developed this model called MOPUS. This is the little logo that we have here. So I'm just going with clicks now. So each click there will be another image come up. And MOPUS basically has two surface stores. So the next click now, there's a surface store and a subsurface store, basically simulating the build-up and die-off and wash-off of organisms in each of those surfaces or each of those stores. We tested this model on, so the next slide now, tested this model on a range of catchments. So in this particular slide you see we've got a prediction versus measured event mean concentration graph, and you can see that the Nash-Sutcliffe efficiency there was around 0.76, which is pretty good. And you see a table here with lots of numbers in it. Don't focus on all of the numbers, just focus on the top line there, which is called EC, which is the Nash-Sutcliffe efficiency for concentrations. And as you can see, for each of these sites, we're getting around 0.2 to around 0.4, 0.5 Nash-Sutcliffe efficiency. And to the untrained eye, some people might think that these numbers are quite low. For urban stormwater quality prediction, though, they're actually pretty good. In typical pollutants such as sediments, you're looking at actually less than that. So you're looking around 0.2. So these numbers are actually not too bad. If you just click one more time, you'll see another table pop up. We applied it to another 6 different catchments at a range of different scales, right down from Raleigh, which is a very small catchment, to the Yarra River catchment, which is a significantly large catchment in Melbourne. And you can see that the numbers there, again, are similar to what we saw before. So the model seems to work okay. Not perfectly, but pretty good for a non-conservative pollutant in stormwater. So we, I wouldn't say we conquered fecal indicator organisms in urban stormwater, but we wanted to move on and start looking at pathogens and seeing how we can use pathogens to better understand the risks of reusing stormwater. So we did a large collaborative grant with 4 states around Australia, and we monitored 10 different catchments with a range of different events and a range of different land uses in different climates. And we monitored these sites and we analysed them for a range of different constituents, and including pesticides, but today I'll just be focusing on the pathogens. You'll see a little symbol down here that looks like Coca-Cola. In fact, it's called the Cave Clan. The Cave Clan is a group of underground dwelling organisms. Unfortunately, the organisms are people. They hang around the caves of Melbourne, which basically are the stormwater drains of Melbourne. So during this project, we had a lot of our little sensors, which is this little blue thing you see next to the man standing there, ruined by, we thought, the Cave Clan, but they claim it wasn't there. So interesting. If you click to the next slide, you'll see a big table with lots of numbers. I'm not going to ask you to read all of those numbers, but mainly just the first row there, just to highlight the different pathogens that we analysed for. So we did a lot of different pathogens ranging from Campylobacter, adenoviruses, Legionella, we did some Staphy as well, Staphylococcus. And these numbers here just represent the percent positive samples in the catchments that we monitored. I'm not going to go into the details of this because I want to focus on one particular case study. So if you just click once more, you'll see a slide which is called Pathogens in Urban Stormwater Troops Creek Case Study. So this Troops Creek system is an urbanised catchment. The purple area you see on your slide at the moment is probably the only highly urbanised area. It's mainly hobby farms at the top end of the catchment. You'll see two blue, light blue shaded areas, one in the middle of the catchment and one down the bottom of the catchment. That's the wetlands, and we monitor the inlet of the downstream wetlands, so the one that's the bottom left-hand corner. So today I'll be just talking about the influent quality of that wetland to represent urban stormwater. So if you just click once more, you'll see two graphs, one with E. coli and one with Campylobacter. If you just click 4 more times, the 4 box plots will show up for you. So you can see on the left-hand side we've got E. coli concentrations. The open box plots are the ones that samples taken during dry weather. The closed box plots are those taken during wet weather. As you can see, E. coli concentrations are around what we typically find in urban stormwater, so around 10,000 per 10,000 to 100,000 per litre, or 10,000 per 100 mil. And wet weather concentrations are higher than dry weather, which is fairly standard. If you click once more, a grey box or grey shadow will come up over Campylobacter, the Campylobacter box plots. That grey shadow represents the range of Campylobacter that we typically find in Melbourne's wastewater systems. And so you can see the Campylobacter that we're measuring in Troops Creek, the stormwater drain, which would only contain stormwater, is having quite high levels of Campylobacter. So you can see median concentrations of greater than 100 Campylobacter per litre. This is culturable Campylobacter, by the way, not PCR. This is culture-based method. And so we're getting numbers up to around 1,000. We've got a couple of 1,500 up there, 1,500 per litre, which is quite high. So if you look into the Australian guidelines for stormwater harvesting and reuse, the maximum concentration that they report in their measured data sets is around 15 mpm per litre. So we only measured less than 15 in about 10% of cases in our study. So you can see that that 15 mpm probably is not representative of all stormwater catchments, particularly this particular site, the Troops Creek catchment. That has quite a significant influence on any quantitative microbial risk assessment that you'll follow, because if you're using a maximum concentration of Campylobacter of 15 mpm per litre to predict risks or to determine treatment requirements for reuse of urban stormwater, you could imagine that if you're using 15 instead of 1,000, that treatment requirement or the risk would be quite significantly higher. So I think Sunny Jiang will be talking a little bit about quantitative microbial risk assessments after me. So we thought, okay, maybe it's just in Troops Creek that we're finding high levels of Campylobacter. So we looked into our previous data sets for a few of the other sites around Melbourne, and as you can see here, so if you just click once more, you should have a blue table that kind of comes up on the same page still. See the numbers of Campylobacter in these 3 catchments are the 95th percentiles are certainly greater than the 15 mpm per litre, and sometimes up to a few hundred per litre. So it's clearly not just one site specific, and this is now, we've done this at another 3 drains now, and we're finding very similar results. So we've got 6 drains in total, 7 drains in total now, finding very high levels of Campylobacter. Why are we interested in Campylobacter? Campylobacter in Melbourne is the #1 leading cause of gastro. So it's a really important microbe to be looking into. I'm not sure if you can see this video, so for those of you who can, then understanding the level of Campylobacter in this wetland was even more evident after our latest field trip. So if you click once more, you'll see a video, I hope. If you can't, then it's a pity, because you'll see people swimming in the wetland that we're monitoring. And those people swimming in the wetland would be exposed to on that day around 500 mpm per litre of Campylobacter. If you put that through a risk assessment model, the probability of infection is rather high. So I'm not sure what happened to these 2 guys, but I would expect they probably had some problems the next day. So we thought, okay, let's have a look into this into a little bit more detail, because we thought, well, are all the Campylobacter that we're finding, although they're culturable, are they actually infectious? So are they actually potentially disease-causing if humans are exposed to them? And so we would love to be able to do another human feeding trial as scientists, but clearly that's not going to happen, so we can't feed these Campylobacters that we find in the environment to humans. That would be not very nice at all. So the second best way is to start looking at potentially looking at the full genome of these Campylobacter isolates. So that's what we did. We started extracting the DNA and sending them off to sequencing, and we've done it for 200 environmental isolates now, ranging from stormwater, river waters. We've also collected animal feces and sequenced them. We're doing them for human clinical isolates as well, obtained from hospitals around Melbourne. And to begin with, what we've done is we've just extracted these 7 housekeeping genes from these genomes and applied what we call multi-locus sequence typing. And we can then use the types that have been identified using this approach and compare them to databases, international databases, to see what or where these isolates have been obtained from previously. So, for example, we get an isolate from the environment, and we extract these 7 housekeeping genes and find out that it's a sequence type A, and we look at that sequence type on the internet and find that this particular isolate has been identified. And this is just a pie chart. If you click again, you'll see a pie chart appear. That pie chart demonstrates that around 60% of the isolates that are in this international database have actually been obtained from human stool samples and human blood samples, clearly demonstrating that this particular sequence type of Campylobacter has potential to cause human infection. We can do that quite a lot, and we can compare all our isolates that we obtained from the environment to all these international databases. And we can, so if you just click once more, 2 more pie charts will appear. And so we found that about 20% of our isolates that we obtained from these sequences were linked to previous human infection, whilst the other 80% were never isolated from anything to do with a human, and were mainly isolated from waterfowl. So that gives us a better understanding about potentially what level of infectiousness these Campylobacters could be. We're also looking at comparing their full genomes together to see whether or not we can extract some information about how closely they relate to full databases as well, but also extracting things like antibiotic resistant genes and seeing whether or not they're prevalent. Interestingly enough, we found that 2 of our isolates from the environment genetically matched with less than 200 single nucleotide polymorphisms differences in their genomes to our clinical isolates, which suggests significant local relevance. And so I'm quite interested in that, especially considering we've only done about 20 clinical isolates so far in Melbourne. So we didn't expect to find 2 already that had pretty good matches. If you click once more, sorry, the text is kind of hidden by the slide. We wanted to do more than just genetic-based analyses, because we wanted to see whether or not these guys could actually start invading into cells and producing cytotoxins. So what we also are doing what we call attachment invasion assays onto human cell lines to see if these Campylobacters can actually invade into the cells in vitro experiments. We're also doing cytotoxicity tests to see if they can actually produce the toxins capable of damaging human cells. And we're doing some motility tests as well, because motility is a function required for infection for Campylobacter. Well, not required, but very prevalent in infectious Campylobacter. If you click once more, you see a big ugly green table, just demonstrating that we're not just looking at Campy and bacteria, we're also looking for other pathogens, so looking at adenoviruses, enteroviruses, Giardia and crypto. I don't want you to go through the numbers there, other than to say that we're finding adenoviruses, enteroviruses, Giardia and crypto during wet weather events, but not yet during dry weather, although there are very few samples so far. These are all culture-based methods that I'm providing. The other thing that we're doing, so if you click once more, you'll see a lot of pretty colours come up with the titles inflow, outflow, waterfowl, feces and sediments. The other thing that we're doing at the moment is just doing some more sequencing to identify other pathogens that might be of potential concern in urban stormwater. And so the bar charts that are represented here basically represent the sequencing that we've done on the 16S gene. We're amplifying the V3, the variable regions 3 and 4, and basically using that to identify down to genus level the different bacteria that might be present in urban stormwater, and utilising that to help us inform what type of other maybe more specific qPCR tests we can do to identify other pathogens of concern in urban stormwater. I wanted to cover a little bit about microbial source tracking, so I hope I have time. If I don't, then you can just mute me and run, like I did before. But I wanted to present just a little bit about microbial source tracking. So for those of you who don't know what microbial source tracking is, the objective is here is just to identify where the microbes are actually originating from. And the reason why we want to do that is because we want a better understanding of risks. So it's a common belief that microorganisms that are sourced from human sources, such as failing septic systems or potentially sanitary sewer overflows or combined sewer overflows, would potentially cause a higher level of risk to humans utilising these water sources downstream than the feces of, for example, cows or wildlife, other wildlife animals, or agricultural animals. So we're really interested in whether or not we can differentiate between human and non-human sources of contamination. And also from a management perspective, so if we understand where the sources of fecal microorganisms are coming from or originating from, then clearly we can have a better chance of mitigating that risk or mitigating that source of contamination than if we didn't know where they were coming from. So in the early days, we started using chemical markers such as fecal sterols, and this is just a nice little graph demonstrating some of the work that we did, but I'm going to skip that now to the next slide. So the slide now is titled Fecal Sterols versus Kefeng. So we used a lot of chemical markers in the early years to see if we can identify sources of contamination, in particular human sources of fecal contamination into urban stormwater systems. And whilst we found there was fairly good inference of whether or not there was human contamination in there, we did start applying these markers more widely to other catchments, and we fell into a whole heap of problems. One is just the caffeine example. Unfortunately, in Melbourne, in urban areas, there's a whole heap of coffee shops. So if you've been to Melbourne, you know that we're very particular about our coffee. I'm not sure why, but we are. And so there's a lot of coffee shops, and unfortunately they dispose of their coffee grinds into waste bins that often don't have lids. So when it rains, we get this caffeine that comes out into the storm sewer. So what we were finding was that if any of these urban catchments actually had commercial areas in them or a few cafes, we had quite high levels of caffeine detection. So that kind of put that marker out into the bin. If you click once more, you'll have a title called MST Bacteroides. So Bacteroides for the last 10 to 15 years has been a commonly used marker, and the idea is that there's certain strains of Bacteroides that are unique to different animals. So there's one here that I've listed in the first bar chart here is called HF183. That's supposedly very specific to humans. Unfortunately, there's a whole heap of evidence that these markers do have cross reactions, and we get a lot of false positives. And in our lab, just as an example, I donated my feces. I thought that was very kind of me. Unfortunately, my feces were analysed for this HF183, and indeed I came up positive, which is good. So it reassured me that I am indeed a human. Unfortunately, though, we also detected HF183 in the cow feces that we obtained, which was a bit of a shame. And in fact, according to the cow marker, there's a ruminant marker that's supposed to be specific to ruminants. Unfortunately, I'm also a ruminant, so that's a bit of a problem. So if you click once more, you'll see a little title come up after my picture that says single hit markers. There's a little bit too much room for error, and so there's a little bit of a work in the literature at the moment that suggests that instead of using just one marker for microbial source tracking, we should be utilising multiple markers to make inferences about the sources of fecal contamination. If you click once more, you'll see again a title that says MST Bacterial Community Profiling. And it's got 3 steps: Step 1, Step 2, and Step 3. The idea here is that instead of using one microorganism to infer a source, what we're trying to do is use an entire community fingerprint to determine sources of fecal contamination. So the idea is that we go into our catchment and we collect heaps of source samples. So this is step 1. We go out and collect bird, human, and cow in this example, and we collect them multiple times, and we sequence the 16S gene, and we look at this variable to region 3 and 4, and we identify the fingerprints of the different organisms that could be potentially in each of these sources. The second step is to obtain the water sample that we want to know where the sources of contamination are coming from. And so we call this a sink. So we go out and grab a sample of stormwater, for example. We do the exact same process, and we extract the DNA, and we go to sequencing, and we find out this profile. Then we use some Bayesian modeling. That Bayesian modeling basically just compares the sink profile to all these different source profiles. And it does it in a way which, in the end, provides us with a nice simple estimate of what the proportion is, or the likely proportion is, of contamination between human, bird, and cow, for example, in this simple example. And we can now apply this, and we've demonstrated the application of this process to a whole range of different urban stormwater systems and river systems around Melbourne, demonstrating its ability to identify sources of contamination. If we were to go back to that Troops Creek example, so click once more, you'll see another blue table that comes up with bat, bird, gull, horse, human, and rabbit. This is using that bacterial community profiling approach on the Troops Creek inlet. And as you can see, the majority of the samples that we've collected here identify human as a significant marker of contamination in this influent, really quite highly contaminated with human feces. Now, these numbers here are taken during dry weather periods, and so they are quite high. We know in this catchment there are leaking septic systems, and so I'm not super surprised by these numbers. I'm probably higher than I expected, but at least we're identifying that maybe some of these Campylobacter are actually indeed human, which is confirmed by some of our sequencing data sets. So if you click once more, you just come to slide 20, which is the conclusions. So fecal indicators in stormwater and MOPUS, we can try to model them. The model's not perfect, but at least it's a first start. Pathogens in monitoring in urban stormwater, they're there. I suppose that's not really of particular interest there to us, but Campylobacter was certainly higher than we expected. But I suppose we have a question about what do they really mean from a human infection perspective. Are they actually infectious? And I think we need to do a lot more work to understand that question. And microbial source tracking in stormwater, can we continue using these one-hit markers, or should we go to this broader, full community scale microbial source tracking, or at least using multiple sources of evidence, multiple lines of evidence to do microbial source tracking? And I think if you click once more, the slideshow is over. So that brings me to an end.
Beautiful, beautiful, David. Thank you very much for that. I think that was a remarkable overview of all the great things that you're doing. You know, one of the things that struck me, and then I want to open it up for broader discussion, was your use of Bayesian statistics to back out the likely partitioning of the sources among the various categories. One of the benefits, I guess, of Bayesian statistics is it also provides some estimate of error. And I'm wondering, those pie charts are obviously their lines are not fuzzy boundaries, but I would be very interested to know, what the likely error is associated with those numbers as well.
Yep, very good question and good comment. So the errors are huge. So yeah, you're right. I didn't show any of the specifics there, but the errors are quite significant, and the process is quite a complicated process to get this source tracking. It's called source tracker, the actual model, to get it working well. And the problem is that the source tracker model has to do what we call rarefication before it does any of those Bayesian statistics. So we have to rarefy the data to a certain level first. So that means basically randomly sampling that big community to begin with, and only sampling potentially 100 of the sequences that the 40,000 that we have per sample, for example. And so, in fact, each time you run the model, you may get a different result. And so we've identified that as one of the problems, and we've looked at the sensitivity of all of these different parameters that are involved in the modeling, in the Bayesian modeling, including the rarefaction depth. So if we just rarefy to 1,000 versus 10,000 versus 30,000, what are the results? And indeed, they do change. As you probably would anticipate, the higher the contribution, so if there's a really strong signature from a particular source, those uncertainty bands get reduced quite significantly. For those sources of contamination which are very minute, which could be, for example, human wastewaters in typical urban stormwater systems, maybe not in Trips Creek, but in others. The error bars get quite significant. You're up to like the 100% range. So you can, if you're estimating a mean of 5%, then you could be between plus and minus 100% of that number. So they can be quite large, and we think that the degree of that error has a lot to do with the number of samples that we have in our database, which is actually quite low at the moment to derive a community profile, an adequate community profile of a particular source. And also the fact that we're only going down to genus level, and those that's really not specific enough to start getting really accurate information around particular sources of fecal contamination. So there's a whole heap of work to be done. But at the moment, yes, the error bars are quite large. I can give more specific examples, but yeah.
Thank you. That's a fabulous answer. Are there any other questions or comments for David?
Yeah, I had one. This is Brandon Winfrey. I was wondering if you had any thoughts on the best type of green infrastructure, or WSUD, to deal with the pathogens in the stormwater. Which ones are better at not just treatment, but maybe just reducing exposure to pathogens in stormwater?
Okay, good question. I actually had those slides in here today. Unfortunately, I deleted them, so maybe I should put them back on. I don't know. Anyway, so I had 10 slides, but it was just too long. So we have monitored a range of LID or WSUD, or whatever you want to call them, green infrastructure systems for pathogen removal. The one that we've been focused on a lot at Monash has been biofiltration systems, and indeed we've had a good collaboration with UCI on doing some work on virus removal in these biofiltration systems. I believe that Anna mentioned the fact that we're trying to incorporate copper-coated zeolites in these biofilters as well, which can also help improve removal of pathogens through these systems. Indeed, we do get 1 to 2 log reductions of most pathogens. So we've monitored these systems for actual pathogens, not just indicator organisms, in optimized conditions. So depending on wet and drying regimes and the optimal design of the biofiltration systems, I think you could push. I think we aim to push them to 2.4 log reduction for bacteria, 1.2 for protozoa, and 2.2 for viruses. And we succeeded with that when we incorporated the copper-coated zeolites into the media. But we're trying a whole heap of other ideas, like using antimicrobial plants, so plants that excrete antimicrobial compounds into the soils to help reduce pathogen levels. Dosing these systems with different microorganism communities to see if we can kind of outcompete the pathogens that are introduced into the system. We want to get around introducing copper into a natural biofiltration system. We've done a lot of monitoring of wetlands. To be honest, I don't believe in wetlands. The wetland that I was speaking about, the Trips Creek wetland, typically produces Campylobacter. We're doing some source tracking which identifies it's probably coming from waterfowl that live within the wetland itself. We're not getting much inactivation, if any at all, through the wetland via irradiation or even settling. So wetlands we're not seeing as very promising technologies at the moment, but biofiltration systems certainly look like a goer.
Great. Any other questions for David?
I have a question if you can hear me.
Absolutely. Go ahead, Steady.
Okay, yeah. Hey, David. Well, I missed most of your slide as your talk because I couldn't load them. Later on, I got downloaded a smaller version of your slide. I'm looking at you have a pathogen in urban water, urban stormwater. There is a table you presented, and there is HPV as a marker. There's a very high rate of positive detection.
Yeah, human polymorph. Yep.
Is that human papillomaviruses in the stormwater? Is that a marker of human sewage?
Yes, that's what they were used. So this is Simon Tozer's group in CSIRO utilizing that. So that's on slide 7 for those of you who got that up.
And then
yes,
all through the.
very high level.
Sorry. Go ahead. I didn't catch.
No, I was just saying that it's a very high level of detection, like we're talking
Right.
about.
Then you discontinue of working on it. Why is that?
Sorry, say that again.
The data for HPV have very high detection level, and then there is most of the Victoria and the West Australia sample was not testing for HPV. Is that correct?
Yeah, that's correct. Yeah, so we didn't test for them at all in those samples.
So those are by PCR method?
All PCR. Yeah, we had a lot of. When I say we, I'm talking about our collaborators at CSIRO. Had a lot of problems with inhibition, especially with the Victorian samples.
Oh. So by comparing those sort of stormwater from all different states of Australia, do you see a common trend and pattern from this?
I personally don't. I don't. My personal opinion is that I prefer to utilize culture-based data, and so that's why I skipped over this as quickly as I could. Because my personal belief is that we should be looking at culture information. I know that you can't do that for a lot of the viruses. So there is a report on this that I can send through. Actually, I could probably make available on maybe the PIRE website or maybe this Google form. I'm not sure how to make that happen. So the guys at CSIRO have created a report on this and looked into patterns, temporal and spatial patterns of all of these indicators or pathogens that they've been looking at. So yeah, yeah, I think the report is probably your best bet to get more information about.
And also to understand the risk and not only seeing positive or negative for pathogen, the other very critical information is the dose, is the concentration. And then we have been struggling trying to figure out how to convert those positive detection into certain type of concentration so that we have a better understanding of the risk. If I do look at your report with the concentration information on it, which is slide number 12, and most of the detection are below detection limit.
Which slide are you talking about, sorry?
Number 12. I think
number
this
12.
is 12.
12. Ah, here we go. Yes. Okay. Yeah, definitely. And we're talking like quite large volumes here. So we're not talking small volumes of water done. So for the crypto Giardia, it's often more than. Sometimes we can only get 10 litres, depending on the size of the event. But often there's more than 30 litres of stormwater that's being filtered.
Is this also PCR-based method?
No. So the slide 12 is all, well, not all of it's culture. So the Giardia crypto is done by the US 1623. Sorry, I forget numbers nowadays. US 1623 standard for Giardia crypto microscopic observations, but we also do a cell line assay for crypto to see if there's any viable crypto there. Adenoviruses and enteroviruses using the A549 cell line to enumerate adenoviruses and enteroviruses using an 11 NPN method.
Right, interesting. The only positive results coming from the adenoviruses in here.
Yeah. It's we have more data than this. This is just for one site, so this Troops Creek wetland. We have other sites or other urban creeks around Melbourne where we've got more data than this.
Very interesting. I think those will be a very valuable data set and for I guess the following up the risk quantification. So thank you.
Yeah. What do you think about the Campylobacter, Sonny? Is this okay for me to ask a question, Stan?
Absolutely. Go right ahead.
Well, the number is quite high, but I think maybe we need to understand a little bit more of those Campylobacter do have the sort of disease-causing capability or not. I think
my hypothesis is that it's much lower than. There's not a lot of good information around on that proportion of infectious. It's very sporadic, and as people say, 20% or something, and just apply a fixed number of 20%, which is odd. But yeah, anyway, it's interesting.
Well, that's great. Maybe in the interest of time, we could transition to the next talk. So thank you very much, David. And next up is Sunny. So Professor Zhang will talk to us about her study. Her quantitative microbial risk assessment study of using stormwater runoff for various purposes. So, Sunny, if you want to take it away.
All right. Do I need to come to live? Am I alive?
I do not see you. So your camera is probably, there's a set of icons at least at the top of my screen. So if you click your camera, there you are. Okay.
Okay. And then I'm going to use this slide uploaded into the Dropbox shared box, because this slide has a set of animation, but when it's uploaded to the Dropbox, I just click through it. The animation is not there anymore. But hopefully, I will be more in pace with everyone else if I'm looking at this side of the slides here. So, well, this is Sunny Jiang from UCI, the home of anteaters, and greetings to everyone. I try to talk about quantitative microbial risk assessment here. I want to start with a very brief overview to guide you through the topic here. I want to introduce briefly what is quantitative microbial risk assessment. More often, you're going to hear this as QMRA. And I will follow up with why is it important to apply QMRA in stormwater harvesting after hearing those viruses and pathogens in the stormwater. Maybe you're worried about the risk. How is the QMRA used in evaluating the risk for stormwater application, and how the QMRA can help to improve the LID management. So the next slide, you will see the outline of QMRA is a 4-step structured risk assessment approach which incorporates scientific data to estimate the microbial risk associated with a specific event. And that can be stormwater reuse, or wastewater reuse, or de facto reuse, or any of the event you're interested. Those 4 steps in here include hazard identification. In this case, we're looking at the microbial pathogen concentration in the stormwater or the water you're of interest. Exposure assessment focus on how much the dose exposed to a human. And then dose response assessment focus on infectious risk due to that specific pathogen. Finally, you come to a risk characterization. This is basically how you interpret the results coming from this type of assessment. If you are compared with a benchmark, do you need intervention or actions? Can you use this result to make decisions? So that's structured step of microbial risk assessment. Next slide, of slide 4, showing why is QMRA important. Most people are scared of unknown. If you are giving them a new type of water to use, such as stormwater to use for shower or toilet flushing, unknown about the risk make people worry. They are more resistant to acceptance. So if we have a good understanding of the risk, that may help for people to accept a practice. They are also needed for decision making. If the water can be used for toilet flushing is safe, maybe you don't want to use it for showering, maybe more risky. This type of a practice can also improving the stakeholders' confidence in accepting those type of practice by letting them know what is the risk. And this type of risk assessment can also provide a benchmark for risk comparison so that you understand nothing is risk-free. The next slide shows how is that being used in evaluating the risk of stormwater applications. And then the left part of the slide you'll see is there's a storm drain. If you look at the storm drain, there's potential viruses in the storm drain. If this type of stormwater is harvested and then being treated by those LID technology and stored and finally distributed for different type of applications you see in that green box, and then that application can expose to people to certain concentration of the pathogen that is not yet removed through those low impact development technology. And then that can potentially expose the person who is, say, using the water in the recreational, playing in the recreational field or the golf course and being irrigated by that type of harvest stormwater. And now if you know the risk, by estimate the risk, and the time is to make a decision, is the risk acceptable? Do we need additional treatment? Or is that feasibility is wise? So that is how it's being applied. And the first step, using an example of stormwater, and you first need to identify what is the important pathogen in the stormwater. Many different type of microbial pathogens are in the stormwater. People normally worried about human viruses because there's a good reason those type of viruses are not easily removed by environmental or engineering treatment system. They can be causing diseases in very low dose. For example, the human noroviruses are the leading cause of human viral gastroenteritis. 90% of outbreak of gastroenteritis can be attributed to noroviruses. It's known as winter vomiting bugs, and then cause people to vomit. Most time those are self-eliminating diseases, and some people don't even have a good diagnosis of that disease. And again, human viruses, such as human noroviruses, are only shed by human. So this picture you see on the right is a image of human norovirus. It's a very, very small particle can hang around in the water for a long time. To get the human virus concentration in the stormwater is not easy. I've been seeing previous presentation, you may be seeing that too. Most of the viral detection are present and absent. Human noroviruses, again, is not easy to detect. Again, there are also no culturable method for human viruses. To get the norovirus concentration in the stormwater, you have to look data from what is published in the surface water. There's many data, you can compile them and try to imagine that surface water is because there is a runoff from stormwater or other source of water. By compiling those data, you can build a distribution curve as you see in this slide. And then by sampling that distribution curve in a random Monte Carlo type of probability analysis, you can achieve a concentration perhaps in your stormwater. That's one type of a scenario. The next slide, if you have a different type of stormwater harvesting system, such as collecting rainwater from the rooftop, in this type of rain collection system, you will not have human noroviruses in there, most likely, because sewage won't end up on the roof. But you may have bird feces and other wild animals make nests on your roof system. This time, when the rain is falling on the roof, collecting in your rain tank, you may have other type of hazard. Those could be including Giardia and Salmonella. They are coming from the feces of birds and other wild animals. Again, you either can collect your own data, looking at the rain tank, or using all those volume of studies done by others looking at Salmonella and Giardia concentration in the rain tank. The box on the upper right corner you're seeing are those concentration range or distribution of Salmonella species and Giardia that have been reported in the rain tank. With those type of microbial concentration data, you can consider how those humans can expose to those microbial pathogens. And most related to water exposure is showing on the left side of this person's symbol. You can get exposure through aerosol transmission if you use the water for toilet flushing or use it for showering. Or you can get exposure through ingestion of contaminated water, perhaps through irrigation of your salad, or irrigation of the tomatoes you consume raw in your own garden. This is showing a scenario of food crop irrigation using rainwater here. So if you have concentration of the pathogens in your rainwater tank here, and you have how those rainwater is transmitted into each different type of food crop you use for irrigation on the small table on the right upper corner, and then you can see tomatoes, lettuce, and cucumber. Each of the different type of crop can retain different amount of water. That again is a distribution. And by knowing the amount of water retained and the concentration of the pathogen in the water, you can calculate the concentration of pathogen potentially retained on lettuce, for example, and then using that concentration of water retained and plus the intake rate of humans' normal daily intake rate, you can get the dose of pathogen being ingested by human by a daily consumption rate. Here is showing a scenario in the following slide, a showering of toilet flushing scenario. In this type of a scenario, your water droplets are being generated by shower or toilet flushing agitation. Those droplets can potentially be airborne. That's called aerosol. There have been reports showing aerosol contain viruses in them. In this case, if you breathe in the aerosol during the showering event, and then you will find those aerosol carrying viruses landing on the infectious site. The bottom of the slide and then showing the dose of the pathogen inhaled is the function of the aerosol concentration, pathogen concentration in the water, and the breathing intensity and pattern of human. So they contain both biological factor, the pathogen factor, and the physical factor of different size of the aerosols, because different aerosol tend to land in different location of human body. That is how do you compute the dose of viruses that ingested by human. The third step of the risk analysis include a dose-response assessment. In this step of assessment is a mathematical relationship between the infection probability and the dose based on epidemiology study. Most of the QMRA researchers were not able to collect their own epidemiology studies. Those are data based on historical hospital data based on the epidemiologists collect those data. But if you fit the data with using the infections risk and the dose, you'll find a function that fit best with that curve that's called dose-response curve. Giardia can be characterized as exponential function, and Salmonella can be characterized as a beta Poisson function, and then noroviruses is the most complex and then can be fit by a confluent hypergeometric function. Each of the functions are illustrated on the bottom of each graph. I hope you can still follow. In the next slide, the final step of risk assessment is risk characterization. It's a conversion of those per event risk or per daily risk into a metric that is comparable to a benchmark. Normally is made by US EPA or WHO that is considered acceptable risk for human use of certain water or application. And then this type of risk characterization from individual event to an annual infectious risk is based on a function of independent theorem. Based on this, you compare to the risk benchmark, you finally can really have a little idea about what is going on. This is how to interpret the result in the next slide. This slide is showing an example of using adenoviruses, noroviruses, and also different type of pattern of breathing when you're showering. Some people are oral breathers. They tend to use their mouths to breathe, and the other are nasal breathers, and then they are more breathing heavily with their nose. And that also determined how many viral particles will land in your lung or in your stomach. And since adenoviruses and noroviruses have different site of infection, those are important factors. What you're seeing here in this complex graph without the animation will be a little bit difficult to illustrate, but you're seeing the red box is adenovirus, the blue box is noroviruses, and then the different level of panels showing the different activity or scenarios this type of water is being applied. Top is toilet flushing, followed by shower with hot water, shower with cold water, and then the final bottom of this box showing food crop irrigation. And then in the middle of the box, you see a vertical line here. This vertical line is the EPA benchmark for annual infection risk of 10 to the minus 4. That's the benchmark. Based on this outcome, you can see most of the risk from the two viruses for toilet flushing and hot water showering is pretty much fall behind, below the risk of annual acceptance, while the other activities, especially for food crop irrigation, will exceed that EPA benchmark. Then what do you do with this result? That's a good question. If you compare this risk coming from food crop irrigation from using secondary effluent to irrigate the food crop, that is another night and day. I didn't include that slide in here, but this is what the comparative risk come about. Okay, how to use the QMRA? It's a good question. This slide, again, it has an animation in this presentation before and identify all that 4 steps you have included in this type of QMRA analysis. The final steps in here for risk characterization. If my risk characterization showing that the final risk is not acceptable, you can have an engineering solution to play into this type of stormwater harvesting. Maybe you can improve the treatment, add a microfiltration before you distribute your water into a household for, say, showering purpose. If you include that 10 log reduction, your concentration will change. That will influence your final risk estimation. So the QMRA can be used as a guideline to see if your treatment is efficient enough to protect human health. Another scenario is to try to change the way the water is being used. And here, this on the left side of the next to final slide, and showing how to use QMRA for LID management. The left side showing a triangle. Our life is balancing between social, environmental, and financial. That's called the triple bottom line. If you want to try to make the water to be usable for reused for purpose, for social benefit, financial benefit, you may want to judge what type of treatment you need, what type of end use you need. If the water is not good enough for showering, perhaps it's good enough for toilet flushing, as you have seen. If not good enough for, say, irrigating of your salad grain eating raw, maybe you can use it to irrigating your orange plant, which is not going to have a good contact with the water. So this is a potential application of the QMRA. This is my final slide here. That's the first trip to Australia. Very memorable. So thank you very much for listening. I guess I can entertain a few questions if you have.
Thank you, Sunny. That's great. Before I turn it over to everybody else, I'm sure there are some questions out there. I was struck by one thing listening to your talk, and that is that, you know, I'm a modeler. I like models very much, but I'm also mindful of the fact that, you know, the old adage that models are all wrong, but some are useful. And I guess one way in which models can be very useful is in identifying kind of the critical pieces of information that we don't know, or we don't know very well, and that limit our ability to make predictions. And I'm just wondering, sort of having gone through this process of really you know, going from estimating, you know, the concentration of, say, viruses in the stormwater, and then exposure, and you know, and then you've got the dose response curves and so forth. You know, so where in your assessment in that chain is the greatest potential for improvement if resources were allocated to better understand that that data set?
Well, certainly, you're absolutely right. Sometimes we very much struggle with the primary data, what is coming into our model. And as a microbiologist, I do feel the concentration of the pathogen in the water is very, very critical. I would say that is the most important factor in it. Most of the report of pathogen in stormwater, as I asked David already, they're present absent, which is very unusable for this type of analysis. And then so that's why we struggle quite a bit with putting into the data. Don't put me wrong, and because I'm a microbiologist, and we also have a tons of problem with the dose-response relationship because the data in there is extremely limited, and there is no good study. Most of the dose-response curve based on a study done with patients about 30 years ago. So, but there's not much I can do about that, and I think most people believe that as well. Well, I think we can do better with the pathogen detection.
Mary Estes at Baylor College of Medicine, I know, did some dose response studies with norovirus, and she said that it was a problem getting graduate students to sign up for her lab because they all had to do at least one feeding study. But anyway, yeah, thank you very much. Are there other questions for Sunny from the panel or other speakers?
I actually have a question. Sort of the trend, as I saw it in David's work, was moving away from single indicators into sort of fingerprints or multiple indicators. And I was wondering if there is a similar kind of way to use QMRA so you don't have to do for just one or the other. You could encompass multiple indicators.
Yeah, it's very, very difficult. And then so we have done that with we just add them up together, but you add them up not just a sum of different risk from different, not indicate different pathogens, right? And then so if you have norovirus, you have adenoviruses, you have salmonella, you have a lot of things in this water, you can do individual risk assessment for each, and then you sum them up by using the theorem of independence. And so you're going to run a huge amount of Monte Carlo in this, and then more you have the burden of running the program is. Yes, you can do it, but we try to limit them to 3 and 4, so that's possible.
Thank you.
Sure.
Are there other questions for Sunny? Excellent. Well, thank you so much, Sunny. What a great presentation. It was super clear and very, very deep at the same time. It was wonderful. Thank you. So next up on our roster is a presentation from Dr. Meg Rippy. Basically, she will talk about an analysis that she did on pesticides in stormwater. So we're kind of transitioning from the microbial risk to the chemical risk. And so with that, I'll turn the screen over to Meg so she can tell you about her work.
All right. So as Stan said, I'm Dr. Megan Rippy. I'm a postdoctoral researcher at the University of California, Irvine. And what I want to talk about today is urban pesticide variability and its implications for our sustainable water future. I'll try to keep track of the slides, but sometimes I tend to forget. So apologies in advance. So moving on to the second slide, I kind of wanted to start out by talking about the big picture, looking at worldwide pesticide use, because I think understanding that big picture helps us understand sort of the problem relative to urban stormwater. So the first thing you need to know really is that pesticides are a big business. There's a global market. They're a very valuable commodity. And because of this, they are heavily used. Indeed, by 2007, we could say that worldwide application of pesticides for agricultural and urban purposes totaled more than 2 million tons at a cost of over $39 billion US, and that's in 2007 equivalents. So it's a lot of pesticides. And where exactly are they being applied? Well, this map on the bottom of slide 2 from the Food and Agricultural Organization helps us understand that. The colors on this map are average tons of agricultural pesticides per 1,000 hectares of arable land that are applied between 1992 and 2010. So white colors indicate low pesticide application, and dark blue colors indicate very high pesticide application. And I've sort of circled in red the areas that are big pesticide users, obviously the United States, certain areas of South America, Europe, China, and Australia. And moving on to slide 3, we see that there are consequences of this kind of heavy pesticide use. For instance, widespread global contamination of surface waters, groundwaters, sediments, and soils. And if we just take a US-centric perspective for a moment, and we look at some information provided by the USGS, the US Geological Survey, we see that more than 95% of streams and rivers sampled by the USGS between 1992 and 2011 contained pesticides, a huge number, and more than 50% of groundwater wells in agricultural areas were also contaminated. And given these really poor statistics, it's sort of a logical question to ask, who's to blame? So moving on to slide 4, we see that traditionally we've blamed agriculture. And that's what this plot by Stone et al. In 2015 is intended to indicate. On the x-axis, we have percent of streams with 1 or more pesticide compounds that exceed a chronic aquatic life benchmark. So that would mean that they are toxic to algae, vascular plants, invertebrates, or fish. And then on the y-axis, we have 2 different time ranges, top being 1992 to 2001, and then on the bottom, 2002 to 2011. And it's clear that a large percentage of agricultural streams in the United States have pesticides that exceed 1 or more of those aquatic life benchmarks, but that percentage is stable, if not declining. So this piece of information is one of a myriad that has led us to move away from just blaming agriculture and to think about urban pesticide use being a real problem. And that's shown on slide 5, where I've added the urban information below the agricultural information so that we can see that pesticide contamination in urban streams is really increasing, moving from 53% between 1992 to 2001 to 90% between 2002 and 2011. And this means that a larger fraction of U.S. urban streams are now impaired than agricultural streams, and really the majority of U.S. urban streams are impaired. So what are the pesticides and the herbicides that are responsible for this? So who are the culprits? And in the U.S., it's generally commercial weed killers and insecticides, so a lot of our lawn care products. And there are a wide range of these, but 3 of the sort of big groups would be the phenoxyherbicides, the triazines, and the polyureas. Phenoxyherbicides were developed in the 1940s, some of the early herbicides. They're synthetic plant growth hormones that kill broadleaf weeds through uncontrolled growth. They essentially grow the plant to death. 2 common compounds are MCPA and 2,4-D. You may be familiar with 2,4-D. It was one of the 2 main ingredients in Agent Orange, and now you can also find it sort of on your shelves as Weed Be Gone. Triazines were developed more in 1950s. And they kill broadleaf and grassy weeds by inhibiting photosynthesis. 2 of the common ones that we'll see are simazine and atrazine. And interestingly, atrazine is banned for domestic use in many countries, an exception being the United States. And polyureas, like triazines, were developed in the 1950s, and they also target broadleaf and grassy weeds by inhibiting photosynthesis. And the key player here would be diuron, which is an herbicide, an algicide, and is often used in anti-foulant paint. So it's really common in the environment because it's used for so many applications. So a logical question to ask now is, does this global story reflect the US story that I kind of just told a little bit about? And unfortunately, we have real geographical knowledge gaps in our understanding of urban stormwater pesticides. We know a lot about the US and a lot about Europe, but many of the other sort of key pesticide using areas we know much less about. And so what I want to talk about for sort of the remainder of this talk is a little bit of work that I've been doing with Australian colleagues to help improve this situation, at least for Australia. So now we're going to move on to slide 9. And this is just an outline for sort of the remainder of the talk, which is really going to talk about a case study of urban stormwater pesticides across the continent of Australia. First, I'm going to talk a little bit about the study area and the stormwater sampling methods, then talk a bit about measured stormwater pesticide concentrations, and I'll compare those concentrations to those from North America and Europe, public health standards, and aquatic ecosystem benchmarks. We'll talk a little bit about dominant pesticide patterns in space and time, and even some possible drivers of those patterns, focusing on pesticide chemical properties and the co-occurrence of pesticides in common use groups. So on slide 10, I'm going to talk a little bit about the study area, which is really the continent of Australia. David actually presented some pesticide data from the, pathogen data from the same study. So what happened is that stormwater samples were collected from 10 catchments that were located in 4 Australian states. 3 of these were in eastern Australia, so Queensland, New South Wales, and Victoria, and the 4th was in far Western Australia. It's actually the state of Western Australia. And it's important to emphasize that all of these catchments were located in urban areas, so sort of 20 to 100% total imperviousness. So that means we're talking about urban stormwater pesticides, not agricultural pesticides. Regarding study design, most of the stormwater samples were collected using automated samplers that were triggered to sample over a storm duration. So that means we have event mean concentration samples. However, stormwater from Western Australia was collected using grab samples due to logistical constraints. And in total, 27 pesticides were monitored using solid phase extraction and liquid chromatography tandem mass spectrometry. And this includes 2 insecticides and 25 herbicides. So moving on to slide 11, I want to talk a little bit about the pesticide concentrations that we observed in urban Australian stormwater. First thing to point out is that only 19 of the 27 stormwater pesticides we measured were detected in urban Australian stormwater, and that included both insecticides and 17 of the herbicides. All of those pesticides are shown along the x-axis of the box and whisker plot on the right, and the y-axis is pesticide concentration in log micrograms per liter. A couple key points about that plot. First, the box widths indicate the number of samples with pesticide detects, so we have more detects per diuron than, say, per Proxer. The whiskers indicate the 5th and the 95th percentile bounds of the data, and all the outliers are shown as stars. So the first point I want to use this plot to make is that overall, Australian stormwater pesticide concentrations were low. They're less than 1 microgram per liter, which is that horizontal line going across the plot. But they could be quite variable, in some cases spanning 2 to 3 orders of magnitude. And getting back to this idea that overall pesticide concentrations were low, and sort of putting it in perspective, if we move to slide 12, we see that Australian stormwater pesticide concentrations were generally below those of other developed countries, including the US, Europe, and Canada. That's those blue symbols that I've included on the plot now. They're the global medians with the 95% confidence intervals. And the exceptions to this are circled in red, which are MCPA, triclopyr, dicamba, and desethyllatrazine, where concentrations were more comparable. Urban Australian stormwater pesticide concentrations were also generally below Queensland public health standards for recycled water intended to augment the potable supply. Those are the green symbols on slide 13. And the only exception to this was MCPA, where 4 samples exceeded the public health standard. And finally, urban stormwater pesticide concentrations were also generally below aquatic life benchmarks for fish, invertebrates, plants, and algae set up by the US EPA. That's all of these red symbols now on slide 14. And the exceptions here were just for 2,4-D, where 1 sample exceeded the aquatic life benchmark for plants, and for atrazine, where 1 sample exceeded the aquatic life benchmark for algae. So really the take-home message of all of this, which is shown on slide 15, is that Australian stormwater pesticide concentrations are surprisingly low, which is good news for the environment, as well as for programs intending to capture and use stormwater to augment potable supplies. However, variability is fairly high and could be a problem for pesticides with more stringent human or environmental health restrictions, things like MCPA, so further monitoring is really warranted. So at this point, sort of on slide 16 now, we've shown that pesticides are detectable in stormwater and that their concentrations can vary over orders of magnitude. So what I'd like to talk about now is whether or not there are any dominant patterns in our stormwater pesticide data kind of hiding within that variability, and whether or not those patterns are primarily temporal, varying by season or year, or spatial, varying by catchment or state. So in order to answer these questions, we use 2 primary methods, which are shown on slide 17. For our first question, what pesticide patterns are dominant, we use principal component analysis to identify dominant pesticide modes or patterns, and pesticide scores, which really tell us about the strength of those modes relative to each sample we collected. And to address our second question, is the primary source of variability driving those patterns, temporal or spatial, we turn to resampling-based statistical models. And the way these work is you sort your data by the variability source of interest. So if we were interested in variability by state, we'd sort everything by state. You shuffle all of your samples within that source of variability, so within state, perform principal component analysis to generate a set of sample scores, and then repeat the whole process 10,000 times. The idea being, if you have strong yearly, seasonal, state, or catchment-specific patterns, you're going to recover your original principal component scores every time. But if you don't, each time you shuffle, you're going to get a different set of principal component scores, resulting in a lot of error in your final resampling-based statistical model. And I'll get back to the results of these statistical models in a little bit. I want to start, however, with our principal component analysis and this question of what pesticide patterns are dominant. So our principal component analysis identified two significant principal component modes that explained about 52% of our observed pesticide variants. And those modes are shown on the plot to the left, with mode 1 on the x-axis and mode 2 on the y-axis. And this is slide 18. Many people have probably seen plots like these. They're called a biplot, but just in case, I want to walk through them slowly. The black vectors that you see on these plots are pesticide loading vectors for individual pesticides, and you can think of these loading vectors as kind of defining your principal component modes. So we have a grouping indicated by the dark blue star of loading vectors for Micoprop, Dicamba, MCPA, and 2,4-D, and these are our phenoxy and aromatic herbicides. Diuron, our polyurea, is off on its own, marked by the teal star. All of our trizenes grouped together, marked by the red star, and our substituted uracil-bromacil is off on its own, marked by the green star. So this suggests that our stormwater pesticide data is sort of well explained in our modes by these four dominant pesticide groupings. So we can think of our mode 1 catchments as being catchments that have high concentrations of phenoxy and aromatic herbicides and some diuron, and relatively low concentrations of trizenes and bromacil, whereas our mode 2 catchments are those that have high concentrations of trizenes and diuron and relatively low concentrations of phenoxy and aromatic herbicides and bromacil. Another key thing that this plot shows us is all these colored symbols, which are sample-specific pesticide scores. And about each of those scores is a bootstrapped 95% confidence interval that tells us the error about the location of that score in principal component space. And the way you interpret these is the further a score is along a given loading vector, the more of those pesticides are in that sample. So if we look at the red circles, those are from catchment Queensland 2, you can see we have high concentrations of phenoxy and aromatic herbicides, but relatively low concentrations of pesticides like bromacil. So at this point, we've identified some dominant patterns in our stormwater pesticide data. What we'd like to talk about now is whether or not spatial or temporal variability underlies those pesticide patterns. And this brings us back to those resampling-based statistical models. We're now on slide 19. And I've shown the results of two of these resampling-based statistical models on the top, the temporal models, with year on the left and season on the right. And they're oriented exactly the same as this data biplot we've just been going through. And you can see by looking at sort of the output of these models that resampling-based models that resolve only year or season-specific variability do not really reproduce our dominant pesticide patterns. There's really large error about our sample scores. It's actually off the chart. It's off the plots entirely. Fortunately, both of our spatial models, our state-specific and our catchment-specific models, better reproduce our observed pesticide variability. Though you can see there's still some large error about the sample scores in the state model. So what this means is that really our catchment-specific model is our best overall model. Indeed, it explained about 91% of our observed pesticide variants in principal component space. And this suggests that our Australian stormwater pesticide concentrations were varying primarily by catchment. And this has important management implications indicated on slide 21, namely that stormwater pesticide contamination is perhaps best managed at the catchment or watershed level rather at sort of broader levels like the state level. So why might urban stormwater pesticide concentrations be catchment specific? We're now on slide 22. Well, one possibility would be that people living in different catchments just use different products. Unfortunately, we have no direct data for this, but one would suppose that if it's true, then our dominant pesticide modes that we identified through our principal component analysis are likely to reflect associations between pesticides that co-occur in common Australian pesticide products. And if we take a look at those associations using a flow diagram on the right, we see that like does appear to be grouping with like, so this may be true. So how do we interpret this flow diagram? Well, the colored circles indicate products that contain only a single pesticide. So for MCPA, we have 173 products that contain only that pesticide. The thickness of the lines connecting two circles indicate the number of products that contain multiple pesticides. So we have 47 products that contain both MCPA and dicamba. And we can see by looking across, that flow diagram that our main pesticide groups are by and large cohesive, but there are a few crossovers, particularly for like triazines and the phenoxine aromatic herbicides. Importantly, those are agricultural pesticide products only registered for use in pineapple or sugarcane crops. So we're very unlikely to see those in our urban stormwater samples. So for me, the take-home message of all of this is on slide 23, and it's really that preferential application of common pesticide use groups really could explain why certain pesticides tend to co-occur within some of our Australian catchments, but not others. However, there are other possible drivers of pesticide pattern, for instance, chemistry. We can look at slide 24 to see this. We found that pesticides separated into the same 4 dominant groups when we categorize them relative to their environmental persistence. It's the x-axis of the plot on the right, and we use fuel dissipation half-life as a metric for environmental persistence here, and their mobility, as indicated by the log of the organic carbon partition coefficient on the y-axis of that plot. So you can see the same major groups here, and it's less clear why differing chemical characteristics would drive catchment-specific patterns, but one possibility is through climate, because climate can impact both environmental persistence and mobility of pesticides. And given the really broad geographical range of the catchments that we're looking at, it's likely that they had very different climate conditions. So at this point, I'd like to conclude my talk. I'm now on slide 25. I hope I've convinced you that pesticide concentrations in Australian stormwater are surprisingly low, which is really good news for the environment, as well as for programs intending to capture and use stormwater to augment potable supplies. And I want to emphasize here that I'm not suggesting that people drink the stormwater, it's more that they use it for potable substitution purposes. We also found that pesticide patterns tend to be catchment specific, which suggests that stormwater pesticide contamination may best be managed at the catchment level. And finally, we detected several possible drivers of the observed pesticide patterns, including catchment-specific application of common pesticide use groups, as well as pesticide chemistry, particularly related to mobility and environmental persistence, which may be linked to catchment-specific patterns through climate. So that concludes my talk, and I'm happy to take any questions at this point.
I have a question.
Yeah.
Yeah, that was an awesome presentation. And I was wondering, so based on this data set, you can conclude that pesticides in stormwater in Australia are low, but could you find any other published studies anywhere in the literature that contradicted this at all?
That would show low pesticide concentrations in Australian stormwater?
No.
Oh, no, that would find maybe high concentrations.
High concentrations.
Specific studies.
For Australia. There are... just in general. Broadly everywhere, there are certainly studies that show high concentrations in stormwater as well as in roof runoff in some cases where people do aerial spraying of pesticides. So there are certainly instances where stormwater pesticide concentrations are going to be very high, and that sort of came out in the comparison between the Australian stormwater pesticide concentrations and sort of US, Europe, and Canada. That's particularly driven by concentrations in Europe, actually, where pesticides have been used for an extremely long time, and they started to ban things when contamination became a problem and other people started to follow suit. In Australia itself, in terms of publications, there aren't a lot that look at stormwater pesticides. There are some unpublished reports out of Kefeng, and a few studies that are starting to look at pesticide concentrations in wetlands and in agricultural areas. But the urban emphasis isn't as strong yet. But the concentration, yeah, and the concentrations vary. In agricultural areas, it's obviously much higher. But in the urban areas, they do identify some of the same sort of problematic pesticides that we talked about, but the other issue is sort of what's measured. Atrazine and simazine tend to be commonly detected. The phenoxine aromatic herbicides people look for less. So that would be sort of the other problem.
Okay, thanks. That was great.
Can I ask a question? I'm interested in have you done any analysis to link the pesticide concentration against the rainfall characteristics?
Rainfall characteristics. That's something that we are working on, but have not done too much yet. I did a preliminary sort of catchment principal, sort of catchment characteristic principal component analysis to try to see if there were land use or rainfall or antecedent dry period kind of characteristics that would separate out the different pesticide groups. And I didn't have a lot of success with that, though there are at least certain land use characteristics that might matter for certain areas. We have one area that's heavily impacted by groundwater, for instance, that has its own signature. But in terms of rainfall, not much has been done at this point with this data, and I did not have a lot of success in terms of breaking everything out by rainfall characteristics.
Okay, thank you.
Any other questions for Meg? Great. So now we transition to, very logically, pesticide removal in biofilters. And for that, we we're lucky to have Dr. Kefeng Zhang talking from Monash University on some work that he and his colleagues have done there. So Kefeng, if you can take it over, that would be great.
Okay, thank you. So today I would like to talk about the pesticide removal in stormwater biofilters. So Megan has already presented that there are some pesticides that existing in stormwater. So we would like to see how our stormwater biofilters remove the pesticides. So moving on to the outline. So today I would like to mainly talk about the removal of pesticide in biofilters, and through some specific experiments. We did 3 major experiments, and I will be introducing this. And also I would like to share the knowledge we have about the surrogate for pesticide removal in stormwater biofilters, and also at last I want to briefly talk about the modeling of pesticide removal in stormwater biofilters. So moving on to the next slide, for my interest, we only tested a few pesticides, atrazine, simazine, and promethazine, which are commonly found in stormwater. And the atrazine, simazine, and promethazine, they are quite similar in terms of their chemical structures and also their physical chemical properties and half-lives. And the glyphosate is a bit different from other 3 pesticides. It has less mobility, and it has lower biodegradation rate. It has lower half-lives. And we expect that in stormwater biofilters, the removal of this pesticide will be mainly through the absorption during the rainfall event, and also there will be slow biodegradation over the dry periods between the stormwater events. So next slide. It shows 3 different major experiments we did. The first is the in situ challenge test we conducted on our Monash car park biofilter. And also back in the laboratory, we did a laboratory colony experiment to understand the breakthrough curves of those pesticides using the soils collected from the car park biofilter. And also we developed the in situ tool, in situ columns, to study the pesticide removal in the field system. So on slide 5, so this is our Monash car park biofilter, and before we did the experiment, we came up with a few challenging conditions that was analyzed using music. So first of all, about the dry period, so we came up with 2 conditions. One is pretty wet conditions, so like we only have a dry period of 10 hours. And the second dry period is 21 days, which is quite challenging dry period as we found in Melbourne. And also in terms of the volumes, we those 3 pore volumes for the consecutive events, which means that 2 events with very short dry period, and 4 pore volumes for a single event. And the inflow concentration, we were also targeting very high challenging concentrations. Challenging concentrations. So we used 2 milligram per liter for glyphosate and 60 microgram per liter for simazine, atrazine, and promethazine. So according to these challenging conditions, we designed 3 3 tests over a month time, around a month time. So test 1 was done under normal conditions with very challenging volume, and test 2 was mimicking very challenging concentration with only 12 hours of dry period. And test 3 was done after 21 days of test 2, in which we also dosed a very high volume of inflow. So test 3 is basically a very challenging dry condition. So we studied 2 different types of biofilters, and one is the sand biofilter with a submerged zone, and another one is a loamy sand biofilter without any submerged zone. So let's have a look at the results. So this table basically gives the outflow EMC concentrations of the 3 tests for those 2 biofilters, and we can see that the biofilters are very good at removing glyphosate concentrations. The reduction could be up to more than 90%. However, in terms of the triazines, the atrazine, simazine, and prometrin, the removal by stormwater biofilters wasn't very good, and its outflow EMC concentrations were almost achieving the inflow concentration. So we still need to optimize these biofilters further to enhance the removal of these triazines. And this is the next slide, slide 7. This shows the intra-event variability of the concentration change over the 3 tests. So this is, I'm giving an example of simazine concentration. So as you can see, in between test 1 and the test 2, there's very short dry period. So from the beginning of the test 1 to end of the test 2, the simazine concentration were increasing all the time without any decline in the dry period. However, after a very long dry period between test 2 and test 3, so we observed a decline of outflow concentration change. So basically means like the starting concentration of simazine in test 3 is lower than the finishing concentration of simazine in test 2. So that indicates that the slow biodegradation of simazine is happening during this dry period. And also comparing those 2 different biofilters, although they are very different in their configurations, but we can roughly tell that the loamy sand biofilter, which is the blue dots, and it has a better removal performance in removing the simazine, as the loamy sand has a higher organic matter content than the sand and filter medium. So this is the in situ experiment. The next slide is shows how we did the laboratory column experiment. So basically, we designed the laboratory columns exactly the same. I mean, in terms of the soil profile as to the field challenge condition, and the inflow volume was we dosed these columns with the pollutant with the pesticides, and then flush it with a certain volume. So for this one, because the glyphosate has a much higher absorption potential than the triazines, so we dosed the more volumes and also used the more volumes to flush the column. And the inflow concentrations were close to the field challenge experiment. And here I want to see like this, the soils collected from the field were sterilized. So in here, in this laboratory column experiment, we were only studying the adsorption process. And the adsorption process will be the main process during the wet period. So moving on to the next slide, I'm showing some breakthrough curves of these 4 different types of pesticides. So for the atrazine and the simazine, because they are very similar in terms of their mobility and their half-life and their physical chemical properties. So you can see the curves are very similar. And after full pore volumes of dosing, the outflow concentration of simazine and atrazine almost reach the inflow concentration. However, as for Prometrin and glyphosate, because they have a bit higher absorption potential, especially for glyphosate, the log KLC value is much higher than the other three. So we can see that, for example, the glyphosate, even after 15 pore volumes of dosing, the outflow concentration still reached only 0.3 of inflow concentration. So the biofilters will have a bigger potential for removing the glyphosate. And from these curves, we can also see that the biofilters wouldn't be very good at removing the atrazine and the simosine. So further work should be done to optimize the media and to optimize the systems for to enhance this removal. And in terms of glyphosate, although it's currently showing that the biofilters are doing a good job for removing glyphosate, but we still need to look at the long-term accumulation of this glyphosate in the stormwater biofilters. So next slide, slide 10. So we developed some in-situ colonies. So instead of doing some field challenge experiment, we want to develop some in-situ colonies that will enable us to do the experiment easier and with low cost. So we were trying to develop a in-situ colonies for our study. So basically, the concept was to insert these in-situ colonies in the field side and collect the water from the bottom of the columns with collection pipes. And so next slide, slide 11, shows the design of this in-situ column and some photos. So you can see from the photo figure 2, so this is shows that there are some holes underneath the column, and we have a chamber sealed those holes on figure 3, and there's also a collection pipe. And during the experiment, we were pumping the outflow water from the collection pipe. However, the pumping rate was carefully calibrated in order to avoid the acceleration of filtration. So next slide, slide 12, shows the results we've got. So the black points shows the in-situ challenge test, as I introduced in the beginning. So that's the results from in-situ challenge test, and then the colored points shows the results from the in-situ colony experiment. So as we can see that the in-situ colonies, they are good at mimicking the trend of pesticide simosine concentration over the 3 experiments. And however, there are some differences. The in-situ colonies always produce slightly lower outflow concentrations than the in-situ challenge test. However, we observed that there are quite different climate conditions in between those 2 different experiments. So the in-situ colony experiment was conducted in winter, while the field challenge test was conducted in summer. And then they also have different antecedent fortnight rainfall and participation between participation between the test 3 and the test 4 for the in-situ colony experiment. But we can still argue that as long as we are using these in-situ colonies under very strict conditions and under strict challenge conditions, these colonies will help us to get some reasonable results we need. So moving on to the next part, we want to talk about some potential surrogate for this pesticide removal in biofilters. So during the in-situ challenge experiment, as well as the laboratory colony experiment, we also tested a range of potential surrogates that could be used for the pesticide removal. So in slide 14, this shows the linear regression between the surrogate and the herbicide remover. So you can see, let's focus on the phosphorus, total phosphorus and the total dissolved phosphor. We can see a very strong linear correlation between the reduction of phosphor species between and the pesticide we tested. And also we found out that the UVA, which is ultraviolet absorbance, is also highly related to the reduction of atrazine, simazine prometry. So those two will be some potential surrogate used for pesticide removal in stormwater biofilters. And we think that because of the similar processes occurring between the potential surrogate and also the pesticides, that the reason that make them significantly correlated, especially for the adsorption process during the rainfall event. And back in the laboratory, we were targeting I'm on slide 15. So this shows the linear correlation between the glyphosate reduction and the total phosphor reduction. So you can see by comparing the field experiment against the laboratory experiment, we observed very similar results. And this basically tells us that this significant correlation between those potential surrogate and the pesticide was due to the similar processes that was accounted for the removal of the pesticide. So the next part on slide 16, I would like to briefly introduce the stormwater biofiltration treatment model for modeling pesticide removal in biofilter. So this was done by one of our colleagues, Anya Randlovich, and she developed this model. And this model has two major components. First is the hydrodynamic component and the pollutant transport module. So if you are interested in this model, you can refer to her paper for more details. And for the hydrodynamic model, we have a dispersion coefficient as a parameter. And the pollutant transport model is a process-based model. We modeled the absorption process using the chemical non-equilibrium two-side adsorption model with the instantaneous fraction of absorption, kinetic parameters such as soil partitioning coefficient, and also first-order kinetic adsorption rate. And the biodegradation rate was could also be accounted using the first-order decay. And I applied this model for my laboratory colony experiment, basically in slide 17. So I'm using this model to calibrate the parameters using my laboratory breakthrough curve experiment. And the calibrated parameters were then used to predict the in situ challenge test under the in situ challenge test experiment. And the next slides, I'm showing some good results of the model application. So for parametering and the graph set, and the dot points is the measured data from the in situ challenge test, while the red lines shows the prediction data based on the laboratory column calibrated parameters from the laboratory colony experiment. And you can see that this model can produce reasonable good results to predict the field performance. However, the uncertainties, you can see the predictive uncertainties, they grew over the three tests, especially at the beginning of the challenge test 3, which was conducted after a very long dry period. So this long dry period introduces a lot of complex processes like evapotranspiration and that biodegradation process that was not very well captured during the laboratory experiment. So come to the conclusion. So we found out that the stormwater biofilters are good at removing the glyphosate remover. However, it's not very efficient for removing the atrazine and the simazine. And also the in situ columns we developed could be a good tool to test the pollutant removal in the field biofilters. And also we found out the phosphor species are a good surrogate for the pesticides, and also UVA are good surrogate for atrazine, simazine, and prometrin. And the stormwater biofiltration treatment model that we developed could provide some good prediction, could be a good tool to predict the field performance of the biofilters for removing pesticide. And also, personally, I think we need to do some feature work to, for example, to optimize our filter media to enhance the simazine and atrazine removal, as the current biofilter media is not able to remove very good, to provide a good removal of atrazine and simazine. And also the long-term performance should be investigated for the pesticides. And also further work could focus on some surrogate reliability under different conditions. So that's it from me. If you are interested in more details of this experiment, you can refer to these papers. Thank you.
Thank you, Kefeng. That was a wonderful data set, and I'm really pleased to see the acronym for your model is MPIRE. So you probably didn't realize that, but now I can take credit, right? Yeah. So I guess we want to go to some questions. I'm sure there are some questions out there, but I kind of wanted to lead off just ask you a little bit about the MPIRE model. So you showed the predictions of the model compared to the data for some contaminant results and breakthrough curves. How does the model perform from a flow perspective? So presumably you start looking at the flow predictions, and then once you've got that nailed down, go to the breakthrough curves.
Yeah. So the prediction results I'm showing here is just the application of my experiment, and this model was was originally calibrated against the field experiment in terms of the flow and also the pollutant. And the flow, because the flow was very well predicted actually, with very high model efficiency. And this is especially for the case because I introduced two different kind of biofilters. And for the biofilter, we send filter media and submerged zone that was extremely well captured in terms of the flow. However, for another biofilter, because that other biofilter was not very well designed, so we had some problems with the modeling of the flow.
Great, thank you. Great, thank you. Are there any other questions for the audience?
I had a couple questions about what you think would be, sort of looking forward, good ways to modify biofilters for removal of trizones, given that atrazine is prevalent in groundwater everywhere.
Yeah. So the first thing on my head will be use some different medium that, for example, biochars and some zeolite, if that's that could be helpful to enhance the pesticide removal. And another thing will be because we haven't looked into the we only tested a couple of systems for the pesticide removal, and we would like to see if the plants, some specific plants, will be helpful to remove the pesticides. So that could be another direction that we can go to enhance this. Thank you.
Thank you. Yeah.
Great. Well, if there are no more questions for Kefeng, I think that we'll move to our last presenter. So basically, we've been looking in detail at sort of what's in stormwater and what that means for human health risk, both for microorganisms as well as pesticides, and then some really elegant experimental studies, kind of at the single unit scale. And then Tim, we are very lucky to have Dr. Tim Fletcher with us, who can basically talk about scaling that up to a whole watershed. So it's a really unique opportunity to look at the microscale processes and the challenges, and then at the end product of how these biofilters get implemented in practice. So with that, Tim, I'm going to turn it over to you.
All right, thank you very much, Stan. It's a pleasure to be here. So I'm assuming my slides are going to come up automatically.
So the way it works, Tim, is that we have shared all of the slides via a link,
yep.
and then
then
when we put this together as a course module, we will we'll merge your face and your slides. But for the moment, we're going to see your pretty face.
I would put more emphasis on the slides than my face if I was you. But all right, so I want to talk a little bit about this Little Stringy Bark Creek project, and in a past one of these seminars, I talked about its role in flood mitigation, and there was a presentation yesterday by Stephanie O'Malley talking about maintenance, etc. So today I specifically want to focus on the water supply aspects of this project. This is a project that has as its central aim to test the question about whether the water quality, the hydrology, and the ecological health of a waterway can be restored by essentially retrofitting the stormwater system using the type of technologies we're familiar with and other presenters have talked about, so rain gardens and rainwater harvesting, etc. But today I'll focus much more on talking about, well, what have been the water supply behaviours and performance of the system. So it's a very large project, been running essentially since 2008. Many people from across the University of Melbourne, Monash University, Melbourne Water, and many other partners. And I particularly want to acknowledge Chris Walsh, who I kind of see as the spiritual leader of this project. So nothing exciting in telling you that urban stormwater runoff degrades streams, and so I'm on to slide, I think it's slide 3, the slide where it just says urban stormwater runoff degrades streams, and there's 2 photos. So a picture of a street and then a picture of a stream. So you might wonder why I'd waste time telling you something you know so well. But it's a really important point, I think, to make that the typical approach that waterway management agencies have had to dealing with degraded urban streams has been to go in and intervene in the stream. So place rock in the stream or try to recreate habitat diversity in the stream, etc. And studies all around the world by people like Emily Bernhardt and many others show that essentially you get no ecological response from doing that. So there's been many studies and meta-studies showing exactly the same thing. And so our hypothesis in this question is, so on to the next slide, we hypothesize that if you want to restore those streams, if you want to, when I say restore, improve their health, then you need to make the stream flow, so the flow regime, the water quality, much more like that of a natural catchment, and that gives you the best chance of that stream then having an ecology which is I suppose desirable or healthy. So we hypothesize that we therefore need to reduce the size and the frequency of untreated runoff going into the streams. We need to restore the quality and pattern of that flow regime, and that flow has to be of an appropriate quality as well. And to do that, we really need to get the runoff volume to match the pre-developed volume. So I'm still on that slide with the tank and the rain garden. And that point about volume is really important because it essentially is saying there is an environmental imperative to remove that water from the stream. And that creates, therefore, a very big opportunity, as we know. So our test catchment is the Little Stringy Bark Creek catchment in Mount and I'll move to the next slide, which has an aerial photograph showing the catchment. So catchment's about 13% total imperviousness, around 9% connected impervious, so there's some informal areas. And so it's a fairly typical outer urban kind of catchment with lots of residential suburbs, sort of lots of residential allotments ranging from sort of 400, 500 square metre allotments up to much bigger ones in some of the areas. It's got a bit of commercial, so there's a picture here of a gas station, and also some industrial. So the last photo on this slide is one of the little industrial estates where you've got lots of things like a concrete batching plant and some panel beating car repair industries and some recycling centres and that sort of thing. So on to the next slide, which is titled 300 Stormwater Retention Systems. So between late 2008, early 2009, and now we've constructed some 300 stormwater retention systems. We'll use that term as a general term. These range in scale from many at the individual household scale, so the allotment scale, through a sort of middle scale of 1,000 to 5,000 square metre catchments. So these tend to be in large systems next to the streetscape, or in the case of the gas station, we've got a very large harvesting system there. We're harvesting some 2 million litres a year from that catchment and using that for a car wash facility. And then through to some very large systems where we are harvesting water from quite large impervious catchments and then supplying regional sports grounds. So on the right-hand side you see some 800,000 litres of storage in those three large tanks. And those have a filtration system, UV system, and then application to the sports facilities you see in the top right. So on to the next slide, which is titled How Well Do the Systems Work? So I just want to talk a little bit about how well they're working from a water supply point of view and how that interacts with their primary, our primary goal of mitigating the stormwater impact. And I'll talk a little bit about their operation, the maintenance and the risk, a little bit about the energy use monitoring we've been doing, and a little bit about their costing performance. So just reiterating this point that in this catchment, so this slide comes from Perrine Hamel, in this catchment, as is the case in any urban catchment, essentially we're dealing with a very large excess of water, and so our whole approach is to try to get rid of that excess and then the remaining water to filter using the type of technologies that Kefeng and Dave and Meg have all been talking about. So in this case we've got about 85% of the rainfall which we have as a resource because that's the excess rainfall that we're trying, excess runoff that we're trying to deal with. So that slide's entitled Volume to Lose, Volume to Use. So I'll click on to the next slide, which is entitled Performance of Household Rainwater Tanks. And so there's a table here which just shows of the some 300 systems we've installed. We've been monitoring quite intensively 12 rainwater tanks, and this is work we did This was done by Matt Burns, a postdoc in our team, and a PhD student, and so he did this as part of the CRC Water Sensitive Cities project on stream ecology. So he monitored these 12 rainwater tanks, looking at their performance in terms of water supply, but also their simultaneous performance in terms of runoff retention. So we've got this dual goal, of course, of reducing discharge to the stream at the same time as supplying water. And again, that might seem obvious to you, but there's some optimization to play with here to make sure we get the best outcome for both of those, and I'll talk about that in a minute. So on to the next slide, which is titled Tank Water versus Mains Water, and this gives a percentage in the dark grey, percentage of the average daily water use that's coming from the tanks, and then the percentage that's being supplied by mains. So the supplier here is Yarra Valley Water, and we have access to the mains water use from each house, and this essentially allows us to see, you know, which houses are using a very large proportion of rainwater for their total needs. So if we have a look, for example, at the household 1, we can pretty much assume that they are using their rainwater harvesting only for outdoor uses, and it would even suggest that their system has failed at some time because there's a very low proportion being used, supplied by rainwater. Whereas if we look at something like household 8, then a very significant proportion is, I'll just turn off my phone, a very significant proportion is rainwater. If we look at household 9, household 9 is using the rainwater for everything. So in fact they don't bring in any mains water anymore. So they have a full treatment system on their supply, and they use our rainwater harvesting for everything. So on to the next slide. So if we think about those households, we see that quite a few of them are not using rainwater for a large proportion of their use. And so if we then look at our monitoring data in terms of the level in those tanks, we see that a lot of those tanks are overflowing quite often. So in other words, when there's a rainfall event, essentially they're discharging to the stormwater system the same as if they weren't there. So that's obviously not a desirable outcome from the stormwater mitigation point of view. So we have been working to develop a kind of hybrid system where the rainwater tank might have an outlet, you know, halfway up or 3/4 of the way up, that supplies passive irrigation to a garden area just through a drip line, essentially, or it might do that through a second tank. So there's various ways of doing it, but essentially what we're then doing is dedicating a proportion of the tank to passive irrigation of open space, whilst the lower part of the tank is dedicated to the what I'll call active uses of that water. So that's a way of improving the runoff retention performance of the systems without any energy costs, etc. These are just passive, so draining under gravity. The question, of course, is, in doing that, do we reduce the reliability of supply of the tank by a significant amount, such that the user of that tank, you know, perceives that it's less attractive to them? And to answer that question, so I'm on to the next slide, which is called Big Impact on Retention Performance. And on that slide, you'll see essentially a cumulative frequency distribution, which is the percentage of time when a given level of retention capacity is delivered by the tank. So I'm just clicking the first animation that shows, for example, in the case of a tank here that supplies toilet and laundry, by adding the passive irrigation, which we call leak here, we probably could have come up with a better marketing term, we increase the median retention performance from about 10 millimeters to about 18 millimeters. So what does this mean? It means that 50% of the time that tank is able to now retain the first 18 millimeters of rainfall before there's any overflow. That's a fairly significant event, so not many rainfall events would be greater than 18 millimeters. And then that passive irrigation essentially drains back down over a period of 2 or 3 days. So from a runoff retention performance point of view, that's giving us a significant improvement. It's getting up to the sort of target numbers we would be wanting. So the question is, how much is that affecting the reliability of supply? So I'll click on the next slide, which is titled No Real Impact on Yield. And so here we see a similar type of plot, but in this case the y-axis is percentage of demand supplied, and you'll see that in the two cases, so toilet plus laundry or toilet plus laundry plus hot water, in each case where we add this passive irrigation, which we call a leak, you'll see that it has a very, very small change on the reliability of supply. So in other words, we find that we can optimize the system to maximize the runoff retention performance with very little impact on the user's benefit received in terms of the amount of water and their reliability of water. Remembering, of course, that all of these systems have automatic switch over to mains, so they don't notice necessarily that the tank is not supplying water, but still the aim is to minimize their potable water use, so that's why the optimization occurs. All right, on to the next slide, which is called decentralization of risk. So one point to make is that we're doing quite a lot of investigation in terms of the reliability of these systems, and we find that these rainwater tanks, where they're operated by a householder, have a non-negligible failure rate, so something around 20% seems to be common, that in any one time there's 20% that might be partially or completely malfunctioning. So for example, pump might have stopped working, or the switchover might have stopped working, or the inlet to the tank might be blocked, or various things. And we've actually been working with the retail water authorities on some kind of new business models that would see rainwater tanks actually installed, supplied and operated by retail water authorities, so the retail water authorities becomes a provider of not just selling water through pipe systems, but they supply water services, including tanks. And that way, when you structure the model that way, you have a much more guaranteed level of maintenance, because you have a regular maintenance. When the meter reader comes to read the meter, they also check the tank, etc. So several times a year the tanks are checked, and telemetry systems make this a bit easier. In terms of at the other end of the scale, the more centralized systems, there's obviously normally a lower risk of failure, because there's more disciplined maintenance regime in place. Having said that, that's not always the case, and we see cases where the risk of failure might even be higher, because the institutional capacity to maintain these systems isn't always there. But the other point is that these large systems, the impact of the failure is greater. So some interesting trade-offs to play with there. All right, on to the next slide, called energy use comparisons. So one of the questions we asked ourselves is, how much energy are these household rainwater harvesting systems using compared to, for example, the potable water system. And if we look at this table, or look at the, actually probably best to look at the graph down the bottom, so you'll see that Melbourne's potable water system before desalination was put in place has a very, very low energy intensity. It's one of the lowest in the world, so somewhere around 0.11 kilowatt hours per kiloliter, so really impressively low, a gift of our topography, I suppose. We're sitting supplied by surface water coming from the uplands. However, the production, obviously, of water using desalination is a completely different game, and so we're into much, much higher energy use. And in fact, this plot shows the annual carbon dioxide emissions rather than the energy use, but of course they're directly related. If we look at the rainwater tank systems, there's two things we notice. The first is it sits somewhere in the middle, so it's significantly higher than the gravity-fed reservoir system of Melbourne, but it is significantly lower than the Melbourne desalination cost. And I should say the desalination cost is just the cost of the energy cost of desalinating the water, not the cost of transporting that water up from the lowest point in the landscape. So in fact that number is very conservative, and it's likely to be significantly higher. But we do notice that in the rainwater tank systems there's significant opportunity to influence the energy use intensity by the choice of pump systems, and we have done a lot of work on measuring the performance of different pump systems under different use patterns, etc. And for example, so we see a number there of kind of approaching 375 300 and somewhere close to 400 kilograms of carbon dioxide there, but that can be reduced by more than 50% just by the specification of efficient pumps. All right, next couple of slides. So I'll skip through the slide called costing. I just want to make the point here. So on the next slide, it's a graph showing the impervious area treated and the cost treated. So dollars, so thousands of dollars per 100 square meters. So just want to make the point that the sort of mean cost is around $1,500 per square meter, per 100 square meters. We've got some projects that actually cost less than zero because the value of water returned by them is greater than the cost. 2 points to make. First of all, the natural instinct all of us would have would be that there would be some economy of scale, so we'd see the cost significantly reduce with bigger systems, and that's not what we see in reality because of the bigger contingencies and bigger sort of setup costs of big projects. So I'm often surprised how often I hear at conferences, economy of scale, and fair enough to say it if you've got empirical data, but in most cases, people are just sort of assuming that's how it is. The empirical data we've collected suggests that that's not the case. One thing I will point out here is that these costs are all for retrofits, so these are retrofits of existing households. We've done some costing on greenfields, and of course you get very different numbers. You get numbers shifted down substantially so that the life cycle cost over even 10 or 15 years is in most cases going to be negative. All right, on to the next slide, which is a quick life cycle cost analysis. So if we take a mean cost of $1,600 per 100 square meters, so that's a $4,000 cost for a 250 square meter roof, which is average there. At 30-year life cycle with a 5.5% discount rate, which is pretty typical for Australia in this current decade. That is about $240 a year per household. This is based on 2011 costs of water when we did this analysis. That life cycle cost essentially ends up zero if the water price doubles, and we are a fair bit of the way there already, so we're kind of getting close to that point already in 4 years. The other point to make, I suppose, is if we think about that as a cost of dealing with stormwater, so $240 a year, the mean wastewater charge in Melbourne is some $500 a year. So it actually makes dealing with stormwater look like a fairly cheap thing, and that is partly because of the water supply benefits that are accruing. Okay, next slide on to conclusions. So just to make the obvious point that we see a nice synergy here between the waterway health benefits and the water supply benefits. The energy use of these rainwater harvesting systems is significantly greater than gravity, obviously, but very substantially less than desalinated water. Costs around 50% of wastewater disposal cost, which is attractive, but with the increases in water costs that have already occurred and are forecast to occur in the next 5 years, we essentially see these systems ending up with a negative life cycle cost. So in other words, a benefit over sort of 20 years. In the case of greenfields, we see that over much shorter time frames, where we see payback over around 8 years, depending, of course, on the context. Okay, and I'll leave it there. Thanks a lot.
Thank you, Tim. That was absolutely fabulous. I think we may need to mute because I'm getting a serious echo. Maybe this will help. So I wanted to lead off with just a quick question, Tim. I was thinking about system-level risks. You know, and your talk really leads nicely into that. Like, for example, one of the unintended consequences or issues that is being faced in Southern California as water conservation begins to happen is that wastewater flows are much lower than they were designed for, and that is creating a situation where some of the wastewater treatment plants are looking at some real engineering challenges, even moving the waste, and you know, issues with odor and you know, just functioning of their systems. Do you see? I mean, you know, you had a case where one of the households went to 100% rainwater. You know, and obviously some of that is going to go to the wastewater treatment plant. But do you sort of see in this experiment some sort of surprises in terms of the system level functioning of the overall water flow through Little Stringy Bark Creek?
Okay, yeah, thanks, Stan. Yeah, that's a good question. So perhaps specifically in relation to that that wastewater risk, we've actually been working with the Retail Water Authority, Yarra Valley Water, quite a lot, and we've been quite pleased with their encouragement and support for the project. And in part, of course, what we are doing is encouraging people to still use water as they need, so not to necessarily, you know, be pushing water conservation very hard, because there is a great amount of water available. So rather than using less water, we're saying use different water, use this rainwater that's available to you. So in terms of wastewater discharge, the households in this catchment are probably not seeing the reductions that might occur in other areas, because we're instead providing them with this additional resource. But in more general terms, that system level risk question is a really important one, and we have a new PhD student who's trying to understand this question about how do you manage the risk at different scales in terms of maintenance, etc. And I think there really is some good work to be done in new business models that address this. So as I say, in this retrofit context, we went in and we've put rainwater tanks in on individual allotments, and then the landholders become responsible for them. In the ideal world, that's probably not the model I would use. I would have rainwater tanks be supplied by a supplier like a water authority, or I'm going to call them a water services authority, who then monitors them using telemetry, etc., so they can tell when a tank fails because the telemetry is saying, hang on, it rained 10 mil, but the water level in the tank didn't change. There's something not right here, so it sends out a little alarm saying you need to go and inspect that one. And it could be a hybrid where it sends a little text message to the householder saying it looks like your tank is blocked. So I really think some of these system level risks and how we deal with them is an area ripe for some applied research. And I think particularly some of the cheap sensor technologies and communication technologies that now exist really place us in a position where a lot of those kind of barriers are easy to solve. I know Dave, for example, Dave McCarthy has been doing a lot of work with those Arduino sensors, and those sort of things become the very cheap basis of a solution to a lot of these problems.
Thank you, Tim. Just as a kind of follow-up to maybe make my question a little more intelligent, because I realized that you weren't really encouraging conservation here in retrospect. It does seem like it poses a risk to the business model for the water suppliers. I mean, if this really does take off, and you kind of hinted at an alternative business model, which is that the water suppliers essentially shift their focus from centralized supply to decentralized supply. Can you comment on that a little bit?
Yeah, that's a good point. So it's a risk in two ways. It's a risk in a direct way in that, all right, they're going to sell less water. It's also a risk in Victoria because of, or in fact most of Australia, because of the structure of the way wastewater is charged, and that is that you pay you pay your wastewater discharge cost essentially as an estimated proportion of your water use. So it's assumed that, for example, 70% of your water use must be discharged to wastewater, and so therefore you pay according to that. That's a problem, because if you start using rainwater and all of a sudden your water use drops, then your wastewater charge also drops, even though you might be discharging just as much. So that's a problem. But you're right, we see that for a range of reasons it would be good to explore alternative models where water, I'm going to call them water supply authorities, become water service authorities. Now, a good example is in the power industry, electricity industry. You all of a sudden see some of the big electricity suppliers in Australia saying, if we don't get onto this solar panel and battery technology, it's just going to take over us. So you now see them offering, we will essentially sell you a solar panel and battery system that will allow you to use less of our power, and basically we'll give it to you for free, in fact, and then you can pay us back over 10 years, and we'll maintain it, etc. So they're recognizing that either the market is going to wipe them out, or they're going to evolve into the new market. And we think the same opportunity exists for water authorities to become a supplier of services that source all types of water rather than just supplying the normal potable.
That's fabulous. So I noticed we have lost quite a few of our panel because it's run a bit late. Can you stay for a few more minutes? Can I just cover you with questions? And I think Brandon at least is on the line as well. So,
yep,
so we'll have a nice private minute we'll have a nice private dialogue. We save the best for last, right? And actually, it's kind of cool because you have that systems level perspective that I think that most of us. I mean, there's very few people in the world who would have that systems level perspective, and so I'm just kind of curious to get your response when you see these talks looking at the microbial risk and the QMRA and the pesticides, and the degree to which biofilters, for example, remove pesticides, energy consumption, and accusations that have been leveled in the past, certainly that the these LID approaches are very energy intensive, and even ecological risk. We heard Andrew Marion sort of talk about, like, for example, we start going toward ornamentals. Does that pose a risk to invasives and so forth? You sort of surveyed, the really big picture. What do you see as the dominant issues that keep you up at night?
Yeah, it's a really good question. I mean, the first thing is, I suppose we work across some of the big picture stuff, but also in the little, I'll call it the little picture. I'm not sure if that's a good term, but I think it's really important that these kind of details are being resolved, because they're the sticking points. They're the ones that when you go, it's all right when we're talking to the already convinced, the already converted, but when we go and talk to groups who are kind of perhaps a bit stuck in the traditional ways, let's say, if we don't have each piece of the jigsaw worked out, so if we haven't thought about the microbial risk, if we haven't thought about the risk in terms of biological diversity in the city, etc., then they're the things that are going to catch us out. So I think it's really important. In terms of the big picture, I suppose I really do see that we do have to get the business models right, because I see, for example, I suppose I'm really excited to see the transition in the energy industry, because at least in Australia I see it's a real dinosaur industry relying on our cheap coal, not wanting to change because the cheap coal is so profitable, and yet all of a sudden the new market comes along and basically says, well, either you change or we're just going to wipe you out and change it for you. And so you now start to see them changing. And I kind of feel like if we can't create that situation in water, we're always going to have troubles. If there's a monopolistic business model that relies on the traditional sourcing of water, and there isn't the opportunity for competitors to that, I think we're always going to be relying on kind of goodwill and getting government to push environmental outcomes, and I don't reckon that's the ideal way. I think we have to have there be a market. So I suppose in the end the economic stuff really is probably the thing I wake up most about, but I kind of feel like all of that can only successfully happen if all the detail is getting filled in the way that the presentations we've just seen are.
Great. And from a regulatory perspective, do you have worries? I mean, I know certainly in the U.S. the issue, or Southern California, I should say, the issue of cross connections looms large. So when you start bringing water into individual homes, that's a big concern. And there may just be, a rule out there or a way of doing business in the regulatory environment that basically blocks innovation in this area. So you mentioned the business model. Do you think in Australia you have maybe more openness in terms of regulatory change, or is that still is that an issue there as well?
No, it's absolutely still an issue. And you've seen in say southeastern Australia attempts to change the regulatory environment sort of gently, and they've tended to be blocked by vested interests, I suppose. And then you've seen attempts to change it more aggressively, and those have created created more enemies than friends, and so even those who might have agreed have tended to become enemies because of the style of. And so I think we still have a lot of significant regulatory barriers, and I know there are many groups, so I certainly try to contribute to resolving those, and I know people like Anna and Dave are contributing significantly to those. But no, I reckon there's a long way to go there, and I suppose that's also why I maybe why I feel so importantly about the market, because I tend to think that changing regulation will be difficult if there are significant players who are against it, whereas if those who care about the environment and those who can see themselves making money out of this all are pushing for that regulatory change, then it becomes harder for governments to resist. So I think, they have to go hand in hand, but definitely I think we do still have a lot of regulatory barriers, and the ones you just cited are absolutely the same ones going on here. I was in a meeting yesterday where they were just talking about using swales to distribute water through the landscape in the school, and the kind of state government people were saying, we don't know about this, we've never heard of this idea of swales, some decades after they became sort of standard practice and hundreds of years after they were invented by the Romans, well, thousands of years, sorry, and they're saying, no, that children will drown, etc. And so the people who designed them said, no, we've calculated the maximum velocities, etc., and these are the standards. And then the state government says, well, how do we know, whether those velocities are true or not? And at that point, I was sort of there as an independent expert, and so I said to them, you realise that the formula, in fact, used to calculate that velocity in the swale is the same formula used to calculate the velocity in the pipe, and you said you'd be perfectly happy if it was a pipe. And it really struck me that there's still, even in government, and this is the Department of Education, you'd think they'd be sort of wanting new and innovative. There's still a lot of resistance. So no, I reckon we as researchers need to keep contributing to that and keep, helping to create confidence that, hang on, this is well established. There's lots of work being done underneath to make sure all this is, the risks are manageable, etc., etc. Yeah.
That's great. So rather than dominate the conversation, Brandon and Meg, do you want to ask any questions, or do you have any thoughts to share?
Yeah, I guess I had kind of a broad question about scalability of some of not just the systems but the studies, and maybe particularly Little Stringy Bark Creek, how the kind of not just business models but maybe even ecological benefits seen from something like the activities of green infrastructure establishment in Little Stringy Bark Creek, how that could be applied to a more urban setting where we're not really looking to get, particularly in LA, we're not really looking to have a huge improvement in the LA River from an ecological standpoint, because we're not really tearing down this trapezoidal channel. There's some restoration efforts, but beyond providing recreation and aesthetics for people, those ecosystem benefits aren't really, we're not really looking to see those in the form of reducing the urban stream syndrome from a hydrological perspective. So I'm wondering if some of those lessons from Little Stringy Bark Creek, how do you kind of envision those being translated to a more urban setting, even in Melbourne? Thanks.
Yeah, great question, Brandon. In fact, I feel like there's quite a few questions embedded in there in a way, or at least there's a few answers I can see embedded. So the first point, I suppose, is that the benefits in Little Stringy Bark Creek, our central benefit is a kind of ecological response of the stream. That's what we're pushing for. But in doing so, we see delivery of a substantial water supply. We see quite significant flood mitigation benefits, which we presented at a previous forum, and you were there. And so it may well be that a water authority or waterway management authority wants to address some of those things, wants to address flooding or wants to address water supply, etc., wants to start to improve the aesthetics of a stream without necessarily being able to get much ecological gains. And they could still use the kind of community involvement techniques that we've been using to get those outcomes. So I think in that sense it's very transferable. The other point is that ultimately our real long-term goal is not about restoring this one kind of degraded stream. It's about demonstrating that this LID concept works, and if it's able to restore a degraded stream, it goes, I think, pretty much without saying that it could be used to protect streams that aren't yet degraded and are about to have their catchments developed. And that's really what we're pushing for, that we're trying to develop stormwater management standards and guidelines based on what we've done that say, if you've got a catchment and you're about to develop it, then apply these standards, and that way you can still have urban development, you can still have lovely suburbs to live in, but you don't have to degrade these streams. You can keep these streams so they keep providing all the ecosystem services. If we then think about the long-term trajectory of quite degraded streams, as I say, we might be wanting for those just to reduce flooding or to improve aesthetics or whatever. But I think we should also think about this in a slightly longer term way. All right, each of us probably only lives 100 years or less, but that doesn't mean the world stops when we do. So if we think about, in Melbourne at least, around 2% of a given catchment or area is renovated, rebuilt every year. It's an amazing number, actually. So on average, every surface in a catchment is somehow renovated, rebuilt within 50 years. So it means that if we think about over significant periods of time, we could actually imagine if we put in planning and regulatory requirements that said whenever you rebuild a road, whenever you rebuild a building, you use these stormwater standards that require stormwater retention, etc. You can imagine that over 40, 50 years, you now have a catchment that's got a flow regime and a water quality capable of supporting a healthy stream. Now you're then going to have to go in and do works to fix up that stream, the actual direct physical works. But it's now going to make sense to do it because you will have taken away that kind of catchment impact. Now, to me, sure, it's not going to be something. If we think about some of those, quite degraded inner urban streams, it's not going to be something where I see the benefit in 40 years or 50 years. Well, no, it's not. I'm not going to see it in 10 years, but in 40 or 50 years, there may be the benefit. But I think we need to make a lot of decisions about, let's say, the Earth's future that are over that time frame. When we make cuts in carbon dioxide emissions, same thing, we're not going to see the benefits in 2 or 3 or 5 years. Our aim is really, in my case, my aim is to see that my granddaughter in 60 or 70 years can still live in a world that she's able to live in. So I suppose I see all of those kind of transferable messages. I think there's kind of many of them. But yeah, you're right, we shouldn't get hung up on this is just a project about restoring one degraded urban stream. I think it's much more about processes you could use to fix a whole lot of stream problems, and much more about setting some standards for future development and future redevelopment.
Fantastic, thanks.
I have sort of, I guess, a comment/question that's for everybody. It seemed that sort of listening to all of these talks, that we need some kind of framework that incorporates and evaluates risk and uncertainty related to risk across multiple pollutants of concern and multiple kind of in-user goals, whether that's things like storage, harvesting, whatever that may be, so that as we move along, we can incorporate what are now unknown unknowns into some sort of living adaptive model for risk. And I guess what I'm asking is, does anybody know of anybody who's doing this? If so, who and where would we start if it isn't currently being done? I don't know if there are any answers to this question.
My only answer was going to be to say I don't know of such a thing, but I really agree with you. I think again, these are the things that we need to kind of give confidence, not just to those who don't want to pursue these ideas, but we also need those to give confidence to ourselves, because a colleague said to me recently, there might be many things wrong with our current water management system, but their ability to supply us water without making us sick is an incredible achievement that we should not give away lightly, and I think that's a very true statement.
Yeah, well, maybe with that, we'll call the symposium to a close. Thank you so much, Tim, and to the entire panel and set of presenters. This was a really interesting set of presentations. Very, on the one hand, 30,000 foot, so we get the big picture, but then there's a lot of fine scale detail there too, which is really fabulous. It speaks to the high quality of all the research that's being done around the table. So and thanks Tim for sticking it out to the end. Ultimately these course modules are going to be set up so that anybody can click on these talks in any order, and I predict yours is going to be the first that people look at. But anyway, it was really wonderful to hear your perspective in particular, and get your kind of intergenerational view, long view of what you're doing. I think that's it's inspirational and very sweet. So thank you, Brandon, to sticking it out, and Meg, and we look forward to the next one. So thank you very much, Tim and Brandon.
Yeah, thank you all.
And thank you, James, for putting it together.
Yeah.